Let me do that one more time. I want to. Hello, everybody. Just getting set up. Give us a moment. Welcome, everyone, to the 2025 Annual AWG Meeting. We're just getting things dialed in. Hello to the YouTube stream. Okay, that should be work well. That's not going to work well. I was trying to have my second avatar. Hi, everyone. Welcome to the 2025 Annual AWG Meeting. We're streaming live in case you need to be mobile. We realize, and thank you for everyone's time. We have a very proud of today's event, and starting off, what we're going to do is first do a bit of a welcome introduction presentation and make sure everyone feels comfortable with the various different features of this platform, and sort of just a general welcome. So I'm Ryan Scott. I'm the John, are you highlighted to speak? I think you are. I just want to double check and make sure where's. Oh, I can do it over there, hopefully. Yeah. So where's the? Oh, good. And then there's John. I'll spotlight him. Okay, so we both should be able to speak now. So I'm Ryan Scott. I'm the point of contact for OSDR for all working group members, and thank you so much to all AWG members as well as the various special guests that are joining today. And John, did you want to introduce yourself?
Yeah, thanks, Ryan. Welcome, everyone. My name's Jonathan Galaska. I'm the acting OSDR project manager, and I'm very happy to have you all here today. Thanks.
Yeah, so we do have a code of conduct, which our colleagues are going to put into the chat if you want to go visit. You know, we want everyone to feel welcome and encouraged to speak up, to engage, and, you know, free of harassment, and so that we can advance science and have discussion scientifically about from data mining to space flight biology, space health, and everything between. So, but we do expect all attendees today to adhere to the code of conduct. So please review that when you get a chance. Today's event, you know, we have I think there's about 20-ish different presentations across talks, as well as the poster session. So we're excited to hear from AWG members' research, learn about, and if you're newer to the AWG, or if you're, you know, just curious about it, you can hear about the various different activities of the groups, how they function, the various opportunities, and hopefully have a discussion amongst each other on potential collaborations, ways to do data mining, et cetera, et cetera. So we'll do this brief introduction, followed by a keynote talk from Dr. Catriona Jameson, followed by another keynote talk from Drs. Michael Lowry and Mirko Prezioso. Then we'll be hearing from the European Space Agency and the first and second place finalists for the 2024 Space Omics Hackathon they had last summer. So that'll include Rodrigo Coutinho de Almeida and Michael Ufemi, Cesare Marino, Claudio Rosell. Then we'll have a break, and then we'll come back and hear from a few of the new AWGs that have started this year, the brain, the hardware, also planetary protection, and of course physical sciences informatics, who are building their own AWGs, which is quite exciting, our sister project. Then we'll have a series of science talks across the AWGs. We'll have an hour for lunch break, and then we'll come back for a second session for science talks, and followed by poster sessions. So before we get too much further, I'm going to hand the mic to John for a few of these slides. But I do want to say on your yes, Sam, it's all actually being streamed live on YouTube as well. So there will be a YouTube link that we'll upload later. But on the main screen that you're looking at right now, if you see in the top right corner, there's a little box. If you go to that top right corner, you can actually change the settings for, do you want to see all the speakers? Do you want to have a grid set up? Or do you just want to sort of, how it's set right now, a carousel view? You can also go to the bottom right and look at your agenda. There's actually a calendar that will take you to that session. And you'll also notice an info board where my colleague Jennifer is posting various links, various different content that's worth people's time and reference. So yeah, and people are free to turn on their camera if you like. I always enjoyed cameras being on. And yeah, so we're going to hear first a little bit from John as the acting project manager for the NASA Open Science Data Repository, and then I'll get a little bit more into the analysis working groups, the data, all the different science, and then the rest of today's meeting. So go ahead, John.
Great, thanks, Ryan. So yeah, again, everyone, thanks for being here from around the world. I'm really excited to hear about what you've been doing. I just really wanted to welcome you. It's a very exciting time right now for biological and physical sciences. A while back, we received an updated decadal survey from the National Academies, and BPS is now looking to respond to that. BPS has a number its overarching missions are to advance the Moon and Mars missions, to enable space exploration, and to benefit humanity through the research projects that it funds. And in direct response to that decadal survey that was released, BPS has now defined a set of goals that it's going to be working towards. So these 5 are on the bottom there. They include precision health, space crops, quantum leaps, foundations, and space labs. This group is perhaps going to be most interested in precision health, which is interested in understanding, the total response to spaceflight from in the animal model, and to understand how we can use that information to improve the health of astronauts moving forward, as well as space crops. The overarching goal there is to start to develop processes and strains of crops that we can use during exploration missions, of course, all based on fundamental science. I wanted to also point out space labs, which is going to be developing a set of tools and processes to allow us to do science better within the space environment. The physical sciences informatics working groups may be interested in foundations particularly, which is really interested in those foundational physical processes that occur in space. And this is important for fire safety, also for fuel management as well. And there's also quantum leaps. Quantum leaps is interested in using the space environment for understanding fundamental quantum processes. So again, this is all a very exciting time. We have these new goals. We have new platforms opening up, including the Artemis effort. And one of the things we want to emphasize is that we really believe that these working groups are going to be an integral part of reaching all of these goals. The output from these groups has been remarkable. We talk about how important these groups are all the time, and are looking to kind of supercharge the outputs moving forward. Next slide, please.
Yeah, just to add on to that briefly though, John. So for the new especially for the new AWG members, I know there's about 10 to 15 talks and posters alone from the AI/ML AWG. So think about those for what types of applied research or operational research can some of the work and the research going on be applied towards. Same thing for the plant AWG or others towards the other section. So, go ahead, John.
Yeah, and so then I just wanted to send our appreciation for your appreciations. This post that was started, I believe, by Marie on the AWG forum, which I hope you're all involved in, was greatly appreciated by the team. You know, as I pointed out, we're in a very exciting time for BPS. It's also, there's plenty of challenges out there. And so to see this outpouring of appreciation for what what the working groups, you know, this what you have built and the amount of support that individuals have gained personally, the personal impact of these working groups, but also the larger OSDR project was really impactful to all of us on the team, and just wanted to send our appreciation for that. So with that, again, welcome. Really glad you're here. We hope that this meeting leads to some great collaborations, and look forward to all of your great work moving forward. Thanks, Ryan.
Thank you, John. Yeah, that post was very touching and was real food for thought and from the heart that the team heard loud and clear. So thank you to the AWG community wholeheartedly. So I wanted to start to talk about NASA Genelab, NASA OSDR, in a broader context of history. I'm one of my favorite slides. I can't take full credit for this slide, but most people probably know Johannes Kepler, his planetary laws of motion, amongst other discoveries, as an astronomer. Less people are probably aware of Tycho Brahe. Tycho Brahe was his mentor of sorts. That was really the last great pre-telescope astronomer. And so because before the telescope was invented, he had spent his career harnessing data collection methods, how to curate his data, annotate the data painstakingly, which then enabled Kepler to come out later to mine that data in the same sense that we think about data mining today, and to come up with a number of discoveries. And so I like to sort of think about this era that we're in for spaceflight biology and space health, space medicine, similarly. As other examples, more recent as well, the Nobel Prize last year with the DeepMind AlphaFold, right? But that was also trained on decades of data from the Protein Data Bank and others to mine that data, to be able to train the tool through DeepMind and others to have that Nobel Prize last year. And in that light, that's how I see NASA Genelab, which then evolved to become, the Open Science Data Repository. Similarly, right, to collect and curate and annotate that data, which leads to fundamental basic scientific discoveries, from kidneys to cardiovascular to integumentary to plant biology, microbial systems, to the applied science and countermeasures, to drug treatments, to various different other countermeasures. Also operational support and mission planning, to have the types of enabling Earth-independent operations, right? So as we go beyond low Earth orbit, how do we, train the systems for biological and health systems for deep space? And so if people are newer to the Open Science Data Repository, it's really a combination of these three repos, in a sense. The Ames Life Sciences Data Archive, which is tasked with everything from physiological to bioimaging to telemetry, like O2, CO2, relative humidity, temperature. It could be CT scans, it could be ultrasound, it could be images. And then Genelab, which sort of pioneered this open science work across the biology and NASA, which is largely concerned with omics, molecular genetic data. And there's also the biorepository NBISC, the NASA Biological Institutional Scientific Collection. And together that forms OSDR, which has a number of different tools. It's got this whole ecosystem. The data has been exquisitely curated to be mineable. And now that we have such good data, there's actually applications and tools to help people do bioinformatics or to do analysis and visualizations to analyze the data more easily, which lowers the bar to entry to have more people contribute and more new ideas come about. Just because sometimes a lot of people might not be too familiar with data, I always just like showing the data, and so it's not too abstract. For example, histology of liver from a rodent, or a complete blood cell count from human astronauts, or biofilm from an ISS experiment, microbial life, to video of Drosophila that flew on the ISS. Just a few of the examples of these hundreds of data sets that we find in the OSDR, the Open Science Data Repository. And this community here actually started really having a discussion on how do you standardize metadata and processing pipelines for omics some years ago. And so, and hundreds of AWG members have contributed to the standards, both on the metadata side, like the investigation metadata, the sample metadata, the assay metadata, but also the data itself, not just the metadata, of course. So this could be tabular data transformations. If anyone's worked with tabular data, you know, sometimes it can be messy. It comes from disparate sources. And so the repository goes to great lengths to standardize all this data so that every single spectrophlorometry results data set is exactly the same, so it can be mined identically. And then, of course, the omics processing pipelines from RNA-seq, metagenomics, single cell, amplicon, and we also have the APIs. The newest one is the biodata API, but there also is a RadLab API and 2 other APIs coming soon. And one of the reasons we've spent all the time has been spent on standardizing all this data is now you can have these different tools that you can, and if you haven't just spent time perusing the data, I always try to encourage new AWG members just to visit the repository, thumb around looking at the data to try to maybe find some interesting questions that maybe a machine learning model that you could actually run the data through to maybe come up with different results or new approaches for something. So, and that sort of brings us to the analysis, the AWG. So the quote unquote AWG is truly 12 working groups, right? So animal, multi-omics, microbial, human, hardware, RadLab, plants, AIML, ALSDA, female repro, brain, planetary protection. And you'll see here the chairs of each group. And if you're newer to the AWG, it's quite important that you contact these chairs. It's their group. They're helping coordinate everyone within it to organize monthly meetings, and the monthly meetings are quite important. So if you're newer to the AWG, make sure to contact the chair to make sure they know that you're on make sure you're on their radar. Maybe introduce yourself at the next meeting, and anyone is welcome to join as long as you are interested in and agree with the 2 main purposes of the working group. One is feedback on standards, right? So like I discussed on the previous slide, feedback on both the data ingestion standards, but also like the data mining, the egress of data, right? So like oftentimes I'll go to the AIML, AWG, or some of us will, and we'll ask them for the best practices to make the data AI ready, or to make the data as highly structured and engineered so that there should be no data cleaning on the egress, right? The data should be as usable as possible. So that's one main purpose of the AWG, and the other main purpose is the collaborations and data mining, right? So I think we're up to about 30 to 40 papers, peer-reviewed publications have come out of the AWG, but in total there's about 150 peer-reviewed papers, preprints, and student theses of the world mining OSDR data collaboratively. So, or just out, around the world. So it's open data, it's maximally open. There's been tremendous growth. You can sort of see a snapshot of those publications here by domain. Actually, in May, I kept following the literature, and I realized that of all the world's literature, there's 35 papers that use AIML for space biology and space health. 19 of them mine data from OSDR, which is amazing. AWG members span over 50 countries, which is profound. There's been this profound growth in the AWG membership, especially since the AWGs are now part of NASA Citizen Science, which is excellent. And then, one thing we've heard from a recent workshop in 2023 was to make things more transparent. So we invented and sort of set up our own forum space, which has a chat function, which has like a post forum board where people can post things. It's a centralized place where all sort of like a Slack or WhatsApp or whatever, where centralized documentation and links can live. Of course, then the calendar, meeting recordings could be centralized. And yeah, so we're pretty happy with how the forum space is working out. We've had just under 500,000 visits to the forum space, which is amazing, since May of 24. Just in the last 7 days, we had over 300 people chatting within. And here are the admins within the forum space, which cut across all 12 AWGs. 11. The planetaries, one we'll talk about later. And also, if you're in the AWG and you're getting a lot of emails, I'm just mentioning this here. Go to your preferences in your profile, and what you can do is please set up your profile picture, make it as professional as possible. And also in emails, you can select how many emails you'd like to receive. I actually just have it set to no emails, and I just make sure to check in once a day-ish, right? Okay, so today's meeting. This is the lobby. There's also this help desk here in the middle. You can use, as you would, up, down, left, right, or WASD if you want to navigate. We also have the YouTube stream link because sometimes we hit capacity on how many people can view this live in the auditorium. So that's why we have the YouTube stream, which seems to be working. Oh, someone's mic is open, huh, Catherine? So the keynotes are going to be in here. The European Space Agency Hackathon talks, the new AWG talks, and Science Talks 1 and 2. We're going to be highlighting speakers, so like Dr. Jameson, Dr. Lowry, and Prezioso. I'll be highlighting you and controlling the slides, and I'll be controlling all slides in today's meeting. And so I'll be highlighting people to speak. There's also the information board there, down at the bottom, which actually has links. You'll see people have already been having different chats. You'll also have the schedule, so you can actually click on the schedule to actually show you where to go. You can edit your profile, and yeah, please make sure to mute yourself and turn off video. If not, well, actually I like video on. I don't know, it just makes it a little more engaging. But yeah, so the help desk, we'll have people around there. If there's any questions or something completely different from the meeting, go to the help desk, and our teammates will be there to help you. Yeah, so if you know other people that want to join, this is a bit of the information on how to join the AWG. This is a snapshot of all the various different meetings, right across over 1,000 people. You can imagine it. We do our best, and thank you for everyone for your patience. We realize the forum space is imperfect, and we're always trying to improve it. So, yeah. So with that, let me end that screen share. Hi everyone. Dr. Jameson. Let me find Dr. Jameson. There we go. Hi, Dr. Jameson. I've highlighted you. Did you want to turn on your camera and?
Just trying to turn it on. See if that worked. Hello there.
Oh, wonderful. This is wonderful. Thank you. Give me one. John, I'm trying to untoggle your highlight. Let me see if I can do that real quick. Was A, B, C, John. Oh, the glass. There it is. So, oh, you already did. Okay, perfect. So, and Dr. Jameson, I did already test out your slides, so I'm really excited for your first slide.
Thank you.
I'm going to share my screen real quick. All right.
All right.
Let me find, oh, there it is. Sorry about that. Give me a second. I know yeah, there we go. That's the wrong screen. Yeah, so hello everyone. Please welcome Dr. Catriona Jameson, MD, PhD. Professor of Medicine at UC San Diego, Director of the Sanford Stem Cell Institute, the Komen Family Presidential Endowed Chair in Cancer Research, also the Chief of the Division of Regenerative Medicine and Deputy Director of the Moores Cancer Center. Her work targets cancer stem cell biology and ADAR1-driven RNA editing, translating discoveries from bench to clinic in leukemia and related malignancies. Dr. Jameson's team pioneered space-accelerated testing of the ADAR1 inhibitor, rebexinib, using patient-derived tumor organoids on the ISS. How cool is that? Her space portfolio spans NASA, SpaceX, CRS flights, SpaceX 24, 25, 26, 27, for stem cell aging studies, plus Axiom's 2, 3, and 4 missions. Quite amazing and inspiring. She recently received the ISS National Labs 2024 Compelling Results Award in Biology and Medicine for the translational findings on ADAR1 blockade. Dr. Jameson trained at University of British Columbia and completed bone marrow transplant fellowships at UBC and Stanford before joining UC San Diego. So thank you so much for being here, Dr. Jameson. We're really excited and honored.
Thank you so much, Ryan, and I really look forward to be able to get feedback from the audience there. This is an interesting way to navigate, new for me, just like it has been in space over the last 9 missions to the International Space Station, where we've really focused on stem cell pathways, aging, and cancer evolution. So, of course, you see that spells space. So our lab is undaunted by difficulty, fortunately, and Ryan's playing the launch of Axiom 4, which was really exhilarating. And hopefully there's sound with the launch because you can hear the excitement in the background.
8, 7, 6, 5, 4, 3, 2, 1.
And there you go. So the reason we were so excited that that launched is it was delayed for almost 3 weeks. And we had to bring our payloads back, reset the nanobioreactors with tumor organoids, fresh samples from patients who were kind enough to donate for research. So we were thrilled that the set of experiments that we worked so hard on with Jessica Pham, our mission specialist in the entire lab, actually launched. So I'll be showing you the data from Axiom 4. We've done Axiom 1, 2, 3, and 4 to look at tumor organoids. And what you may have seen in the news recently is we just published a paper on stem cell aging in space. It seems to be accelerated. So that was covered by CNN, NBC, Reuters Press. You never know what the media will be interested in, but I think the most important thing that you were alluding to earlier, Ryan, is to be able to share the data in an open source way of doing things, considering that these data is important for ground as they are for space. So with the next slide, you'll see what we've been working on for the last number of years. We published this review in the New England Journal of Medicine, which really referred to how we understand stem cell aging and how stem cells go awry and lead to precancer. So what happens with our blood stem cells is that they can acquire mutations that make them differentiate abnormally and actually pick up additional mutations that give them survival capacity and ultimately expand a progenitor group of cells that then learn to self-renew, or in other words, clone themselves when they shouldn't do that. So I'll focus on one thing today, and that's base deaminase deregulation. So what are base deaminases? These are the base editors. So you know about CRISPR, that's the molecular scissors that can cut and replace big stretches of DNA. Base editors act as a molecular eraser where they can erase out a DNA or RNA base. Now that is great for therapy. However, we have endogenous base editors, both APOBEC and ADAR enzymes. ADAR1 has been on our radar for over 10 years when we discovered that unbridled activation of ADAR1 actually leads to precancer evolution to cancer. So I'll be talking about how space has informed our understanding of how ADAR deregulation leads to cancer evolution. Next slide, please. So first I'll start with the APOBECs. APOBECs are mutator enzymes. They evolved to protect us from retroviruses like HIV and to prevent retroviral integration into our genome. Humans have 7 APOBEC3 enzymes. They're unique to primates, and this just shows how important it is to protect our genome from retroviruses. Now, the interesting thing is they protect our genome from human endogenous retroviruses and prevent them from integrating into our genome and leading to genomic instability. So 50% of our DNA is comprised of repetitive elements, including human endogenous retroviruses and long interspersed nuclear elements or lines. They can be activated under conditions of stress, and they're repressed by both APOBEC and ADAR deaminases, meaning they deaminate a base and prevent that integration of the human endogenous retrovirus or line element. So why does this matter for cancer? Mia Petlia, Gruben Harris, and Michael Stratton showed a few years ago in a Cell paper that in over 1,000 cell lines and 577 xenografts, there was episodic activation of APOBEC, and that led to C to T DNA mutagenesis. So normally you think of DNA mutagenesis as happening in response to radiation. This isn't radiation induced. This is stress and inflammatory cytokine induced, and it really important for human stem cell biology. Next slide, please. So we started looking into the APOBEC and ADAR deaminases, as I mentioned, more than 10 years ago, because we found ADAR, which deaminates adenosine to an inosine, actually is disrupted as precancer stem cells become cancer. And it's really ADAR-P150, the splice isoform that is activated by inflammatory cytokines and stress, that we see going up during cancer evolution. APOBECs seem to be more important for precancer. So we're stepping back from cancer and really trying to catch cancer before it becomes cancer and really target it at the precancer stage by focusing on APOBEC enzymes that deaminate a cytosine to a uracil. And as I mentioned, there are 7 of these. Importantly, APOBEC activation can lead to ADAR activation. It becomes a bad Bonnie and Clyde show where these two genomic instability enzymes actually work together to enhance the ability to become cancer and propagate cancer. Because they prevent retrotransposon expression, we actually started to look at these repetitive elements of retrotransposon, human endogenous retrovirus, and line elements. Next slide, please. So what are retrotransposons? So retrotransposons are these repetitive elements that have LTRs or no LTRs. They're sequences that invaded our genome thousands and thousands of years ago. Human endogenous retroviruses litter our genome. They may be important for telling stem cells what they're supposed to grow up and become, but when they're activated for too long, they activate a C-gas sting response, inflammation that leads to interferon activation and interferon-stimulated genes like ADAR and APOBEC. And the same happens for line elements. If you just click again, Ryan, you'll see. So what happens is retrotransposon activation triggers this interferon response that leads to ADAR-mediated double-stranded RNA editing and repression of retrotransposition, where there's a cDNA made that goes back into the genome. Next point. APOBECs also do this. So this was part of a seminal paper that was published in Science by Sean Morrison, where he showed that in mice, pregnancy stress induces retrotransposon activation and actually leads to stem cell proliferation and increased red blood cell production, which of course is a good thing under conditions of stress like pregnancy. But what we know is that can lead to unbridled ADAR and APOBEC activation and ultimately lead to cancer. Next slide, please. So we started to look at space, stress, and longevity, whether or not space-induced activation of these base deaminases and human endogenous retrovirus activation could actually set the stage for precancer initiation. Next slide, please. And this was predicated on the NASA twin study, which was this pioneering study done thanks to Scott and Mark Kelly working together to establish what long-term exposure to microgravity does. So as we know, Scott Kelly was in space for almost a year. He came back with immune dysfunction and pre-malignant changes in the blood, including translocations and inversions in the immune cells, as well as increased inflammatory growth factor expression. So those inflammatory growth factors are the very growth factors that actually activate ADAR and APOBEC. And that's when we decided to start the Integrated Space Stem Cell Orbital Research Center study with NASA. So this was way back in 2021 when we did the first SpaceX mission, NASA funded, and that led to SpaceX 24, 25, 26, 27 to look at stem cell aging under conditions of microgravity compared to ground control, and ultimately led to the study of astronauts, space-associated stem cell hallmarks of aging, SASHA study with Axiom crew members over Axiom 2, 3, and 4 missions, and then the tumor organoid missions, Axiom 1, 2, 3, and 4. So I'll go through all of those. Next slide, please. So how did we study this? So my dad was a coal mining engineer, and he used to talk about taking canaries into a coal mine. Of course, this predated his time, but what miners in the old days used to do is they take canaries in because they could sense carbon monoxide, and if the levels were too high, they would die. So carbon monoxide is something you can't see or smell, but, they had the canaries to detect this. Subsequent carbon monoxide sensors that we all have in our homes basically obviated the need for canaries, and this is what we've done with our nanobioreactor system together with Space Tango, our implementation partner. So what we've developed are these little stem cell nanobioreactors in pediatric blood bags, where we took this gas permeable pediatric blood bag, put a surgifoam sponge inside, put the stromal cells in there that we've irradiated as soil for the seeds, the stem cells, and then we labeled the stem cells with this fluorescent reporter of cell cycle that we made a number of years ago. So if our stem cells are red, they're sleepy, they're happy. Our stem cells should be asleep 80% of the time to maintain their full function. If they're splicing too much, thanks Ryan, they go from red to green. And this is the splicing reporter we're testing right now on SpaceX 33 to see if stem cells splice too much under conditions of microgravity, which would be an indicator of accelerated aging and precancer development. And if they become fully malignant, they turn on this ADAR green fluorescent protein reporter for too long, and we're testing that as well on the ISS right now, SpaceX 33 with Space Tango, in this cube lab, which is essentially an incubator, as you see in the upper right-hand corner there. It has a flask for media, it has an Etaluma confocal microscope on the lid, which when closed goes back and forth on the gantry, and is able to use an AI-generated algorithm to track stem cells in their niche or home and look at stem cell behavior in real time. Next slide, please. So what we saw in these 4 missions that Ryan, you were alluding to, SpaceX 24, 25, and 26 and 27, was we saw increased mutations in space. So initially we thought, well, of course this is radiation. And in doing radiation experiments, we found that only about 20% of the DNA mutations were related to radiation. And in fact, 80% were actually induced by APOBEC enzymes, and specifically APOBEC3C, that introduced the C to T DNA mutagenesis pattern. So what we saw was 5-fold more mutations in space than in ground controls. And of course that concerned us. We don't want to see more mutations in stem cells because stem cells last our lifetime. They're not short-lived cells, they're long-lived cells. So if you get a mutation in a stem cell, that stem cell can clone itself and propagate that mutation. So that could lead to precancer stem cell generation. Next slide, please. So then we started to ask, how is this even happening in the first place? And we started to look into the dark genome, or what my mom calls the Darth Vader of the genome, because we inherit this. Luke, I am your father. So we activate the dark genome under conditions of stress on the ground, but low Earth orbit seems to massively enhance activation of these repetitive elements, human endogenous retroviruses and line elements that should stay repressed during adulthood. They're really important during fetal development, but they should stay silent after that. So you see the orange-red color, that's where you see the change in dark genome activation in flight compared to green ground controls. And that seems to correspond with APOBEC upregulation, both APOBEC3A and 3C. Next slide, please. So this was just published in Cell Stem Cell. This is a high-impact journal where we were able to show the importance of this nanobioreactor technology for looking at stem cell fitness in response to stress and in real time. So we could do this for all of us. We could set up your stem cell nanobioreactor to say, are your stem cells stressed? And should we be developing countermeasures for that? Should we predict, prevent, and reverse stem cell stress? Because if you have protracted stem cell stress, it erodes the ability to make an intact immune system. Next slide, please. So this was covered by CNN, Reuters Press, NBC, The Scientist. A number of groups picked up on this because of the space-accelerated stem cell aging. And what we were really trying to say is it's the activation of the dark genome that can actually be successfully repressed if you put these stem cells onto a healthy stroma or microenvironment. Next slide, please. So when we started to look at astronauts, and this is our space-associated stem cell hallmarks of aging SASHA study in Axiom 2, 3, and crew, 4 crew members, we discovered something very interesting. So we did whole transcriptome, whole genome sequencing, single-cell RNA-seq, and functional analyses before, during, and after launch on these intrepid scientists. And what we found was something very interesting. Next slide, please. We found that there, just like the nanobioreactors, there was a time and space dependent acceleration of cell cycle transit. So you see that on the right. The Axiom 3 crew members spent more time in space than Axiom 2, about 21 days compared to 10 days. Their stem cells went out of dormancy and into cycle, and that accelerates stem cell aging. Next slide, please. But interestingly, they activated that orange population you see there in the Axiom 3 crew members in flight. So we didn't know what that was, and Patrick Chang, who does our computational biology for single-cell RNA-seq, together with Claire Engstrom, said, well, that's a different stem cell population. It turns out that that has massive upregulation of ADAR1 and is a highly regenerative, resilient stem cell population. So we hadn't seen this before. And this seems to come out when you have upregulation of both human endogenous retroviruses and line elements. So you see in flight there, if you just click again, Ryan, there's upregulation, there you go, of lines and HERVs in the Axiom 3 crew more than the Axiom 2 crew. So it means the stress of space activates the dark genome, and this leads to first ADAR activation, and subsequently return to ground, we see APOBEC activation. Next slide, please. And that is what leads to changes in RNA editing, as you see in the top 2 panels there, and in the Axiom 3 crew, increased APOBEC 3C activation. So the reason that matters is that APOBEC 3C activation can lead to what's called clonal hematopoiesis. So this is space-associated clonal hematopoiesis, and you know about the clonal hematopoiesis of aging, where it can predispose not just to acute myeloid leukemia, but also increased mortality from cardiovascular events because it increases inflammation in the major blood vessels in the heart. Next slide, please. So what we saw on the left is the Axiom 2 crew had far fewer clonal hematopoietic mutations than the Axiom 3 crew. So I'm coining the term space-associated clonal hematopoiesis, SACH. So we saw a large number of clonal mutations in the Axiom 3 crew, and we actually got a bit worried until we took the study out to a year. And what we found at a year, actually, that's not on the slide, is that most of those mutations were no longer detectable, meaning something had outcompeted those clonally mutated stem cells, and we think that's that regenerative population of stem cells that is more robust and can take over the niches in the bone marrow microenvironment, so they're stronger stem cells. So that's the resilience piece we were excited to see. This shows you what space-associated clonal hematopoiesis looks like that's induced by APOBEC. APOBEC-induced clonal hematopoiesis, we see this on the ground under conditions of stress. You see the C to T mutation and T to C mutation pattern. When we overexpress APOBEC in aged normal bone marrow stem cells, we see patterns on the right of clonal hematopoiesis, and they tend to be in genes that are epigenetic modifiers or regulate self-renewal, which is the capacity to clone yourself in a stem cell like Notch. So that's disrupted and they clone themselves too much. Next slide, please. So this is what Ryan was clicking on in an earlier slide. So if you click on that, Ryan, what you see is we can track these stem cell populations that have the splicing reporter. You see the red stem cells are starting to give rise to green progeny. So as they splice, the cells turn green, so that's a sign of exon skipping. If they splice too much, you get both red and green cells. So what we see in aging is actually more exon skipping events, and we also see that in leukemia, so more of these green cells accumulate. So what you see in this video is the stem cells track to this niche, this sponge matrix. They love it. They track in there and they settle in there. And that's what we're studying on SpaceX 33 at the moment. What we see when we overexpress APOBEC3C is not only does it introduce C to T mutations, it introduces exon skipping events, and this is a hallmark of stem cell aging and precancer initiation. So we're catching cancer just as at the precancer stage, just as it's getting started. So we see more of these green exon skipping events when we overexpress APOBEC3C in aged bone marrow stem and progenitor cells. Next slide, please. So why does that matter? So the reason that matters is when we overexpress APOBEC3C compared to young stem cells, you get editing of DNMT3A, which is a clonal mutated gene in myelodysplastic syndrome, a pre-leukemic or pre-cancer disorder, and that stabilizes that transcript, leads to enhanced self-renewal of progenitors, this proliferative burst. You see these giant colonies. The colonies are replating or self-renewing more, and then they activate these inflammatory growth factors like JAK2, STAT3. They activate the whole JAK2, STAT3 signaling pathway because of APOBEC activation. So now this answers the question we had from the NASA twin study. Why did Scott Kelly activate JAK2 STAT3 signaling? We think it's because of unleashing of the dark genome that then leads to APOBEC3C upregulation. Next slide, please. So this is what we think is happening. We get dark genome activation under conditions of microgravity and stress. That leads to APOBEC3 inducible clonal hematopoiesis, and that sets the stage for precancer development. Next slide, please. So how do we get from precancer to cancer? This is the last part of the talk. So we know that APOBEC3C induces precancer mutations and RNA alterations, but how does that go all the way to cancer? Well, because APOBEC3C activates inflammatory cytokine signaling JAK2 STAT3, that can transcriptionally activate 8RP150 overexpression. Next slide. So why does 8RP150 matter? Well, this stem cell cloning gene, when hijacked, can lead to cancer's capacity to clone itself and induces therapeutic resistance in over 20 cancers as they invade and spread to other tissues. It leads to resistance that also includes resistance to immune checkpoint inhibitors. So it's a cloaking shield for these cancers because ADAR prevents interferon production in the tumor microenvironment. So the holy grail is to block ADAR P150, and we've made a drug called rebexinib that does just that. Next slide, please. So what we discovered with that inflammatory cytokine inducible isoform of 8RP150 is that it's a metastatic switch. And when that green fluorescent protein reporter turns on, we know the cancer cells, in this case, in a brain metastatic model, are turning on and they're starting to invade. They look like aliens when we have them in our nanobioreactor and also in vivo. We can image ADAR in real time with a green fluorescent protein reporter in space and on the ground and tumor organoids. And what we see is the ADAR positive cells really like to influence the normal stem and progenitor cells labeled in red and actually get growth signals from them. When we look at patients, if you just go back one, Ryan, with lung adenocarcinoma or breast cancer that's metastasized to the brain, we see massive upregulation of ADAR-P150. Next slide, please. And then what we see is the if you look at metastatic breast cancer samples, most of them, all except one out of these 5, massively upregulate ADAR and also these long interspersed nuclear elements and line elements. Next slide, please. So how do you block unbridled ADAR activation? Here we got a bit lucky because to activate ADAR it needs to be spliced into ADAR-P150, and what we're working on was a splicing modulator called rebexinib that just happens to prevent ADAR from being spliced into this malignant isoform called ADAR-P150. Next slide, please. So what we did was we tested this in a leukemia stem cell model, and we found that rebexinib selectively inhibits ADAR-P150 activation. So we said, okay, this looks like it's going to work in a precancerous condition called myelofibrosis and also in acute myeloid leukemia, both of which can confer a life expectancy of less than 18 months. Next slide, please. And if we manage to blockade our P150 based on Nicholas Haney's work, we know we'll be able to increase sensitivity to radiation in solid tumors as well as immune checkpoint inhibitors. Next slide, please. So we decided to test whether or not we could accelerate the development of this drug toward the clinic, and that's rubexinib, by using space as an accelerant for cancer growth. And we got the tip off from Peggy Whitson that this should happen because of work that she'd done before in space. So this is Jessica Pham setting up these tumor organoids. That was correct. Ryan, to skip to the next slide. These are the little nanobioreactors. We've put leukemia in there with a green fluorescent protein reporter, GFP reporter, and also the cell cycle reporter in solid tumor organoids. Next slide, please. So in the Axiom 1 mission, what we saw was tripling in size of the tumor organoids in just 10 days in space compared to ground controls. They proliferated and then they became dormant. The last thing you want is from a tumor to become dormant, because then it resists all therapies that target dividing cells. We also saw activation of ADAR1 in low Earth orbit. Next slide, please. And that ADAR1 was associated with this unbridled tumor proliferation, and it happened in very specific parts of the gene body. So we see this activation of ADAR1. Next slide, please. So then we started to say, can we inhibit ADAR1? As I mentioned, it's inflammatory growth factor activatable. So we tested with both Peggy Whitson and here's Walter Villaday with breast cancer organoids on Axiom 3, where we're testing fedratinib, an inflammatory growth factor receptor inhibitor, versus rubexinib to see which one is the cancer kill switch. Next slide, please. So what we discovered in Axiom 2 is it looked like it was actually not fedratinib. If you just click once more, Ryan, it was rubexinib that started to reduce ADAR activation really early. This is just day 4 into Axiom 2. So we started to see in these metastatic breast cancer organoids that they were very sensitive to rubexinib more than fedratinib. We repeated this. You see fedratinib has some inhibition of breast cancer organoid growth, but rubexinib was the cancer kill switch, meaning if you're not growing, you're dying. The breast cancer organoids just stopped growing, and that was in Axiom 3. Next slide, please. So why were we activating ADAR in the first place? This gets back to retrotransposon activation. So what we see in metastatic breast cancer, and this is not on the ground, this is not in space, this is on the ground, they also activate ADAR and APOBEC, and that combined activation of ADAR and APOBEC actually leads to massive proliferation of the tumor organoids, and this is amplified when we fly these organoids. Next slide, please. So it's amplified in space. So in the last mission, this is coming to the last part of the talk, Axiom 4, we decided to look at metastatic patient-derived breast cancer organoids, glioblastoma multiforme, and say, is the mechanism of action for rubexinib really ADAR inhibition? And certainly for metastatic breast cancer, it looks like that. So this is day 4 into mission. You see rubexinib is already blocking metastatic breast cancer growth on the side, on the left side, and it looks very similar to shRNA knockdown of ADAR. With glioblastoma multiforme, the deadliest brain tumor in adults, they were exquisitely sensitive to rubexinib, but we'd never seen this before. This is space-accelerated biology, and it was actually more potent than shRNA knockdown. If you look at the next slide, here's day 4 on ground. You see the tumors aren't growing as quickly on the ground, but if we leave them for these GBM organoids for 3 weeks, they become massive, as opposed to the rubexinib treated ones that just stopped in their tracks. This is the cancer kill switch, and that's what we've been able to do with rubexinib. So space has accelerated our understanding of how these aggressive tumors really accelerates their growth under conditions of stress. And we have stressors on the ground as well. Next slide, please. So with secondary AML, this is going to be the first clinical target of rubexinib. We plan to treat the first patient here at the Moores Cancer Center later this year. Secondary AML was exquisitely sensitive to rubexinib in flight by day 5. You see there's almost no growth. We transplanted these cells into mice upon return, and it's only the DMSO-treated controls that are showing engraftment. They're still expressing the ADAR nanoluciferase GFP reporter. Those that were treated with rubexinib don't engraft. So in other words, it looks like we're going to be able to prevent growth of AML. This is just going to block recurrence. Next slide, please. So what we've done so far, thanks to our 9 missions to the ISS, has really accelerated our understanding of human stem cell aging, and we've developed tools that allow us to predict, prevent, and reverse the aging process that leads to precancer and cancer stem cell generation. So we're addicted to this space, pun intended. We really want to do more. Space has accelerated the development of this drug, rubexinib, and what we thought was going to be impractical or even impossible turned out to be both practical and possible, and has accelerated our idea of getting to solid tumor clinical trials with this drug. Now we're doing protein crystallization. We'll fly that on SpaceX 34 and hope to get new molecules that are as potent and oral versions of this drug, rubexinib. And I'd like to thank my lab, as well as Axiom crew members, ISS crew members, our funding sources, Aspra Biomedicines that made rubexinib, T. Denny Sanford for his substantial support, and the Moores Family Foundation. And of course, NASA. And the patients who made this possible. I think the point is you shouldn't be able to tell who's a patient in these 2 slides. We want to keep it that way. Thanks.
Outstanding.
Thank you. Thank you so much. I really appreciate you advancing the slides. Clearly, you've gone through these before and you know the cadence.
Yeah, I was just trying to do my due diligence. I was pretty excited. Did you want to make a comment on Starship?
Yeah, so this is Starship. So this is basically a metaphor for space. When we started, we thought these experiments won't be rigorous, reliable, or reproducible, and clearly it took some testing, but ultimately you can do what you think is impossible provided you work with the right crew members. And this is Starship 7. We went to watch this launch. This is when it was caught by the chopsticks, which I thought was pretty dramatic. We were 5 miles away, South Padre Island, but I think we can do more of this. And, we can do this together. We can share the data. We can show where we're all going. Now our data sets are much deeper because of advances in sequencing technology, but also because of the NASA Decadal Survey, where over the next 10 years we'll focus on human biology without having to infer from reductionist model systems. We're having real patients provide their tumors, their blood. This is important for all of us, for our health care going into the future, to predict, prevent, and reverse accelerated stem cell aging, pre-cancer, and cancer stem cell development. So thank you, Ryan. And all of you in the audience, I'd be happy to answer questions.
Yeah, thank you so much, Dr. Jameson. There actually is I think there's a brain viral reactivation subgroup project that AWG is working on. I saw viral reactivation mentioned, and there's also actually a cancer subgroup that they're not presenting today. I think they're in final stages of their manuscript on their data mining activity, but I also hope if any of your colleagues or lab team are around, we're actually going to move to the next keynote. But please feel free to stick around, Dr. Jameson, if you have any time, or I don't know if any of your lab teammates are around. It's sort of why we're having this platform, because you can find one another and have discussions and chats and Q&A on the side. So we're so grateful, Dr. Jameson, and I do hope the AWG can get inspired from this type of approach and what's possible with the spaceflight research impacting health on Earth. So thank you so much.
Thank you, Ryan. I really appreciate all of your work, and then the audience. Thank you so much.
Fantastic.
Have a great day. Bye-bye.
I also believe Jennifer added a number of your papers in the chat, if I remember correctly. Yeah, Wendy, Wendy, if you want to maybe go talk to Dr. Jameson, talk about the brain viral reactivation group and Alzheimer's connection and stuff, that would probably be wisdom. Next up, let me do a time check. Oh, 9 o'clock. Look at that. All right. So let me just change this out. Full screen mode. Perfect. All right. So I'm going to find. Dr. Lowry, I've just highlighted you. If you'd like to turn on your camera, Dr. Prezioso as well. I believe I've highlighted you both. If you want to maybe test your mic and your camera. Hey, Dr. Prezioso, nice to meet you. And hi, Michael. Dr. Lowry, how are you?
Very good.
Should I
Hi, Ryan.
also? I don't have an introduction for Farnood Bayat, but should I invite him to speak as well, or is he just?
No, he's not going to speak
Okay,
today.
okay. Well, but it's nice to have you here, though, Dr. Bayat. So I'm going to share your. All right, so that's showing. All right, so yeah, once again, thanks to Dr. Jameson. That was so inspiring, and I've been sort of admiring from afar, thumbing through your work and research, and so.
Yeah, Ryan, my connection seems to keep going in and out.
Okay. Can you not hear me? Maybe his connection isn't the best. Can you hear me now, Dr. Lowry?
I can hear you now. My connection has indicated that it's not very good. So I think what I'll do, and I'm actually sitting in my office, so you'd think I'd have great connection, and I've got 5 bars, but apparently not.
Okay.
So I'm going to go ahead
ahead.
and turn off my camera.
Wisdom. Yes.
And then just leave the voice there.
Okay.
And so I'll come as a headless.
I will turn off my camera as well.
There we go.
Maybe, Mirko, I'm going to turn off your camera just to help guarantee the connection. So, yeah, Dr. Lowry, so I do have an introduction. So I'd like to introduce you, Dr. Lowry, a PhD who's a NASA senior scientist for software reliability at Ames Research Center. He previously served as Lunar Surface Systems software architect from 2008 to 2011. He was the software lead in the Toyota Unintended Acceleration Investigation, 2010. Also the DARPA chief scientist for the META program on Correct by Design cyber-physical systems for 3 years. Dr. Lowry also led NASA's Autonomy Operating System work, which is advancing neuromorphic computing concepts for intelligent, reliable autonomy in space and aviation. His publication spans software, verification, autonomy, and safety critical systems engineering. Dr. Mirko Prezioso, please everyone welcome him as well. PhD and co-founder and CEO of Mentium Technologies, building mission-critical edge AI hardware. He earned his MS and PhD from the University of Parma in Italy, and worked at Italy's National Research Council in Bologna on spinotronic memristive devices, and later joined UC Santa Barbara in Professor Dmitry Strukhov's integrated memristor based neuromorphic network, which is a field-defining step towards efficient in-memory AI. His current focus at Mentium is dependable low-power inference for aerospace and other high-reliability edge environments. And I'm just very grateful for you two today. And following from Dr. Jameson's, it sort of shows the scope of what the AWG is, and across hundreds and thousands, we have over 1,000 AWG members. It sort of gives a demonstration of the scope of the science, from both the biological to the AI/ML, and sort of helping enable Earth-independent operations. So thank you, Dr. Lowry and Dr. Prezioso. Go ahead.
Okay, so I'm going to talk to you about neuromorphic computing, and
Uh-oh. Lost audio, Dr. Lowry.
okay, I'll repeat that. I'm going to talk to you mainly about neuromorphic computing, which is a real exciting approach to getting high performance computing into space. Neuromorphic means mapping from neuroscience to electronics, and I'll give you a little bit of the history and mainly tell you where it's going. Mirko Prezioso is the CEO of Mentium. I've been working with them since 2016. They're one of our star companies that is helping to advance computing in space. And so let's get going. Ryan, could you please go to the next slide?
Go ahead. Yes, we're on the Viper slide.
Ryan, next slide, please.
Yes, we're on the Viper slide. Oh, should I go to the high performance?
Yeah, so back up one. There we go. So if you look at one of the missions that has been on and off again with NASA Ames, the Viper CONOPS is tethered to the Earth driver. The CONOPS is the rover takes stereo pictures. A ancient RAD-SUM 50 compresses the pictures, transmits them to Earth. It reconstructs a stereo picture for the Earthbound driver. The driver plots an incremental path forward that is then
We lost audio again, I'm afraid, Dr. Lowry.
Okay, so I'll go to the next slide, but if there's an issue, Mirko and I actually did go over these slides yesterday, and if his audio is better than mine, which I suspect it is, please turn on your audio, Mirko. He might be able to take this up. So what the upcoming high-performance spaceflight computer, which is going to be released in 2026, and actually was the subject of one of the first announcements we got from Sean Duffy as he became the NASA acting administrator. It just taped out in July, which means the files for fabrication were sent to a global foundry, and it's going to achieve 70 times the performance, which means that some of the functions of the human driver can be taken over on board. And you can see here some of the specifications. It goes from being able to do 64 bits up to 1,000 megahertz with 10 cores, and it enables capabilities for autonomy that we have not seen before, such as terrain relative navigation, ground hazard detection and avoidance, which is critical as is precision landing, and then on board a surface vehicle such as Viper enable.
Fortunately, I lost audio again. I don't know if Mirko or Michael, you can.
Hi guys.
Mirko takes over.
Michael, this space qualification processor definitely your field. I don't have many.
Okay, I will try to speak. Hopefully my connection works. Once again, a little bit mysterious. I'm sitting in my office, but there is actually no defined space qualification for processors in space, in contrast to aviation. Aviation, if you want to have a avionics that works on, say, a commercial transport, there are certification standards that you have to follow. If you're going to be, say, for a private aircraft, then you can back those off, but there's still certification standards. So space avionics do not have a defined certification process, so you can fly inexpensive and experimental electronics as long as the spacecraft is approved by projects, institutions, and launch providers. So for example, on Test 13, we flew an experimental neuromorphic processor called Intel Loihi together with a desktop, literally $200 microprocessor, but that was approved. Some example inexpensive processors that are CPUs would be the Raspberry Pi and the Android phone, which was put up with
How odd. At least the audio went out again for me. Maybe try again, Dr. Lowry.
Okay, when did it go out?
Yeah, I mean, it seems to last for like a few minutes and then a 20-second delay. At least that seems to be the pattern, but go ahead.
So anyways, the point here is that there is no defined space qualification. As long as you get approved by a flight board that controls the funding and the authority to launch for your mission, you can put up both inexpensive and experimental processors. But in practice, if you're going to go for a Class A or B mission, you are required to meet standards which are defined by the radiation that you expect, space qualification such as temperature, vibration for launch, and that's what is looked at when a Class A or Class B mission is being planned, and a board says that you're ready for PDR. You look up in the NASA Electronics Parts Program what the part that you're considering has been tested to, how well it works, and then you are able to use that as part of your PDR. And we're going to be discussing space computing qualification as starting with experimental and then progressing.
All right, maybe try again, Dr. Lowry. I went to the next slide.
We're going to go ahead and move to this next slide. So the proton flux, which comes from both the solar energy and also galactic cosmic radiation, differs from orders of magnitude, on the order of 8 order of magnitude from sea level up into the unprotected portion of space, which is geo, and of course lunar. And during a solar flare, you'll be getting 10 million protons per centimeters per second. And that not only... Okay, Ryan, it sounds like I'm just cutting out. Perhaps Mirko can pick this up.
I mean, here the point of the slide is to say that there are definitely different scenarios depending on what kind of orbit and what kind of mission you're going to run. And these scenarios, of course, are based on different radiation levels. From sea level to solar flare, there are like 8 orders of magnitude in terms of proton per centimeters squared. Of course, the radiation is not just protons. There are also, and mainly, heavy ions, also coming from the solar wind. And these have different effects on the electronics. You have the so-called single event latch-ups, which are very important. Those are sort of short circuit that happens in the integrated circuit, and then functionality interrupts, upsets in the memory, or different kind of errors that can happen with the radiation. There are two different. Sorry, one more comment on that. You can see there is total integrated dose and single event effects. So these are two different ways of characterizing the effect of the radiation. Total integrated dose means how much the accumulated radiation affects the electronics. So it's a sort of lifespan in terms of how much radiation the electronic can absorb before essentially not working anymore. And then you have the single events. Those can happen from day one and only depends on the radiation that hits the electronics in that specific moment. Maybe we can go directly to this. There are some tools. I believe that's the slide about for characterizing, understanding what kind of environment you will be encountering during the mission. So those are very fundamental to understand what kind of electronics you can actually use for your mission. And then you can use the NEP, which is this NASA part list, essentially, to search for those parts that you believe are gonna survive the environment and for the lifespan of the mission. So there are, of course, different levels of radiation tolerance or radiation hardening For electronics, normally this comes up at a price in terms of performances, efficiency, area, this kind of parameters. What you can do is, of course, testing these electronics first on ground in some facilities like cyclotrons, if you want to test for heavy ions, or proton accelerators. Those can be cyclotrons or even the one in the hospitals. And then, of course, for you can test also for TID, and that is done with cobalt radiation source, with gamma radiation. And then, of course, you want to achieve some space heritage. Of course, that's very important. Essentially, you are testing in the field. You are fielding the ICs, the electronics in space, and see how well they perform. Because, of course, one thing to keep in mind, we cannot reproduce the radiation that you will encounter in space. What we can do is emulate it with the facilities, with different fluxes, and by unshielding the electronics because of the different spectrum composition of the radiation, let's say. Okay, so these are the goals that HPSC and the coprocessor, of course, have set in terms of radiation hardening. As you can see, of course, there is RFI vacuum tolerance, and of course, you have to pass the vibration tests. The temperature range between -20 and 70. The kilorad, those are 200 kilorad in TID, the total integration dose, which is pretty high. Then you have the single event latch-up immunity. So that is characterized by that EV per centimeter square per milligram. Essentially, how much energy you are depositing with these heavy ions into the material of your integrated circuit. And 75 MeV is pretty high. And then you can also characterize in terms of how many errors that the electronics is not capable of correct you will get per device per day into different, or per time, let's say, into different scenarios, which is the Adams 90% worst GEO, and the worst October 98 solar flare. So those are essentially fluxes that have been recorded in the actual places in GEO, and the worst GEO, they call it gestationary orbit, and also during the solar flare that happened in '89, and those were the worst cases that have been recorded. Okay, so let's see what Michael wanted to say here in neuromorphic computing. As he mentioned, it's of course, the name says it's mapping these neuroscience to the actual silicon and computing architectures. It can come in different flavors and ways of doing this. Also very important to say, this neuromorphic computing classification is shifted with time. Normally, of course, you want to achieve high throughput and power efficiency. That's the number one point. You can imagine that our brain, which is the most efficient in many cases in terms of intelligence, it consumes around 20 watts, which is absolutely amazing. And to do something like the brain, if you could, you would consume megawatts, if not more. So the efficiency of the brain architecture is absolutely unmatched, and the idea here is to reach that goal. Next slide. Okay, so the neuromorphic architecture, which is becoming very popular, is the so-called in-memory computing. Essentially, it's with analog computing. Essentially, is mimicking the, if you want, the basic model of a neuron in our brain. What happens is that the neuron in our brain, I'm simplifying this, I'm not, I have no title in biology, I'm a physicist, so please allow me to say this. What it does essentially is summing up the input and is doing a weighted sum of the input over the synaptic strengths of the input comes from synapses. They go into the neuron through the synapses. Each synapse has a certain strength. So we are doing a sort of weighted sum of input by the synaptic strength. So you can mimic that with these analog circuit that you can see on the right, where if your input are voltages, they go through certain resistances that are variable, and you are mapping the synaptic strengths to that resistances, or if you want conductances, 1 over the resistance. The result is going to be a weighted sum of your input by the strengths of those connections. So you are, in a way, emulating a neuron in a very, very simplified way. That was an architecture that Mead from Caltech developed in the 90s, and is actually that the fundamental architecture is used in a number of companies, including ours, as a way to implement a neuromorphic architecture. Please, next slide. I'm not sure, Michael, what was this slide about? Sorry about that.
Actually, I'm not even sure Michael is still highlighted.
Is still connected? Oh, he left? Okay.
He might have.
So let's go directly. I mean, I think this is just a way to map to show the mapping of the neural network, the neuron, essentially what I said before, to that kind of topology that you can see on the right.
Yeah. Here we go.
Okay, examples of aerospace applications for neuromorphic. This is really for AI applications. Of course, you can have many different. You guys know that AI is applied pretty much everywhere at the moment. In space, in particular, you can have it on anomaly detection. Of course, if you want to have some kind of data processing on the different instruments, you can reconstruct 3D images from a single image using the so-called neural radiance fields, NRF. Of course, for autonomy, for proximity operations, robotic repairs, refueling, any kind of maneuvering between two spacecrafts, and of course for rovering. So you want to automate robots on on Mars, on the Moon. When the delay is, the latency is too high, you need AI, and that's the way to move ahead. I don't know if we want to go to this in the discussion, but of course there are different kind of mathematical operations involved in neural networks. There is the learning part, the training part, which is one kind of neural. That's the main message here is this one. The training, it involves some kind of mathematics, which is called the gradient descent algorithm. We don't care that much in this particular case. And the inference, which is the moment in which neural network is used to classify new data. So the how does it work? First, you train a neural network. The neural network learns how to operate on new data, and then you send new data to the neural network to be classified, for example. So that the second part is the inference. And the inference part is a totally different kind of mathematics operations involved, which is essentially mainly these dot products, the vector by matrix multiplication. And that is an important part to understand if you want to understand what makes an architecture efficient. Essentially, for inference, you need to have a very efficient vector by matrix multiplication. Let's go ahead. Of course, there are already examples. This is called heterogeneous computing in practical applications, and you want to have also another way to have reliability is to multiply to have redundancies. That's what happens, for example, in a Tesla. The Tesla has this kind of neural processors, which are actually, again, very efficient GPU. The GPUs are extremely good at running that vector by matrix multiplication. That's the reason why they are used in data centers, right, for AI. And Tesla has their own design for this acceleration of vector matrix multiplication. Let's put it that way. Okay, now these are the slides that I know better. I am the CEO of Main Auditorium Technology, so these are easier for me. I wanted here to give you a little bit of context first of what Main Auditorium Technology does in general, and then how we address the use of AI in space. How do we solve that kind of problem? In general, Main Auditorium Technologies works to solve the AI hardware bottleneck, essentially the lack of sufficient computing power in ultra low power application, in swap constraint, which is the size, weight, and power applications. Satellites are swap constraints, for example, and harsh environment in general. We work to solve that kind of issue, the hardware bottleneck. How do we do that is in the next slide.
You get in there. Sorry about that.
No worry. There you go. So how do we do that? So we are designing a family of AI co-processors. Those are designed specifically for edge application. Edge means not on cloud. Edge means nearby the sensors in the field, okay, close to the sensor, or maybe in your handheld device or body-worn cameras, so forth and so on. These are very unique solutions for different points. First of all, the design that we do, we limit ourselves below 1 watt in power consumption. Currently, we are way below that. We are below 100 milliwatt. But let's say that our designs we give us ourselves in terms of product design that constraint. And that is to get access to the widest possible spectrum of applications at the edge. We have a proprietary architecture that allows the largest model to be run at speed. So that's a very important point for us. We want to run significant models, AI neural networks at real time or faster than real time speed, and deliver accuracy, complexity, and reliability. That's very important for mission critical applications. And we do that by enhancing existing systems. What I mean by that is that we design co-processors. So it's essentially a bolt-on solution that boosts the AI capability of an existing system. So there will be a central processor, a host processor, to which we connect as a co-processor to boost the AI and AI-only capability of the overall system. That allows the end user, the instrument designer, the product designer, to integrate this solution way easier. You don't have to change anything in your software, which is very, very important. Design software takes a lot of time, blood, sweat, and money. All you have to do is connecting our coprocessor and then saying in your software, whenever you want to have an AI inference on some data, just send it to the peripheral. It's like a printer, like printing a document. You want to have a classification on an image, send it to the peripheral, and you will get back the results. And of course, we do radiation tolerant and radiation hardened designs as well. And that's the collaboration with NASA. So essentially, we sell the ICs, the processor, together with the software part that enable the use of those processors. And then also we can sell subsystems in terms of the processors mounted on a specific PCB, printed circuit board. All the applications you can run are there on the left. These are a few classical examples for AI, like object detection, data segmentation, so forth and so on. Maybe we can move to the next slide. Okay, so as mentioned before, definitely capable AI is important for space applications. We are seeing a lot of traction, of course, in the space market, commercial, not commercial. Why? Of course, you can do all the classical AI applications also in space. So you can enhance sensors. You can enhance, for example, you can enhance the resolution of image, super resolution. You can remove clouds from an image. You can remove fog. You can enhance the contrast or low light. You can have real data analytics. That's fundamental because in a bandwidth constrained connection, like happens in space, sending data back to Earth for analysis and then react on that data. If you need the fast reaction times, that is not feasible because of all the delay and the very low bandwidth. And the amount of data is only growing The sensors are getting bigger, the number of satellites is getting way higher. And so what you want to have is a capability to understand what's in the data that you are collecting. It doesn't have to be an image, it can be anything, it can be radio, it can be radar, any kind of signal, so that you only transmit what actually matters, and you can compress that and then send it to ground for further analysis or anything that has to be done with it. And then, of course, that the autonomy in decision making, that's what we spoke about earlier for RPO and all kind of applications. So that essentially spawned this, spawned this long-standing collaboration with NASA that culminated with the delivery of what we call the DVMR. It's not a great name, we're going to change it pretty soon. It's a coprocessor, as I mentioned. This is, and I'm not saying this just because, of course, I'm the CEO of the company, blah, blah, blah. This is a fact. This is the state of the art in terms of efficiency. I'm ready to defend this with any other processor you find around. And it comes, of course, standard grade and rod hardened, which is the one that we are interested in at the moment. Please. So I have a few specs here. I know the audience, not everybody is familiar with the processors and this kind of specs. I'm going to go only on the main thing here. So also some specs are in parentheses. These are for the next generation, the Gen 2, which is coming in April next year. So essentially, the point is, it's int8, so we work on 8-bit precision, not full precision. The size of the neural network that you can run currently is 12 megabytes, so 12 million parameters in 8-bit precision. Next iteration is going to be 16 million. The throughput is 2.4 TOPS, tera operation per second. It's going to be doubled in the next iteration. The typical power consumption, 60 milliwatt for the actual chip. It's going to be doubled in the next iteration because we are going to double the throughput, and also because we are going to add a fast interface. The core efficiency higher 40 TOPS per watt, that nothing comes close to that. For example, if you take the latest NVIDIA Jetson Odin, the one that showed, the golden one, that is around 2 TOPS per watt. Activation memory, so that's essentially the size of the neural net, of the input that you can run. We are going to, now it's 4 megabytes, it's going to be tripled so that you can run full HD images directly into the chip. In terms of what layer we support, so neural networks are made of layers, and what we care about is the compatibility of that single layer. Then you can run any kind of sequence of layers. So essentially, we run 95% of the layers out there for practical neural networks, like the ResNet, the MobileNet, the YOLOs. These are the most famous ones. And the next iteration is going to be able to support also the transformer-based neural network, the attention layer. So for example, transformer-based are very important for visual applications, but they are very famous because of ChatGPT. ChatGPT is a gigantic transformer-based neural network. And this is ready to be connected to the HPSC. So what Mike spoke about earlier. It's a very important program from NASA to substitute that RAD 750 that was in the Viper mission. Okay. So some benchmarking here. You have to do this, of course, if you speak about processors. I have frame per second, essentially the throughput, and I have frame per second per watt, which is the efficiency. Mind that the below graph is in logarithmic scale. So the point is, I'm comparing here our processor with the Raspberry Pi, Jetson Nano, TX2, Google Edge TPU, so the Edge solution from Google, Intel Movidius, and Xilinx FPGAs, Xilinx processor essentially. I'm on 4 different neural networks, which essentially are different kind of layers and in different sequences. So the point here is that, of course, we are way faster than anybody else. We are way more efficient than anything else of their orders of magnitude. But also the important part here is that you can see that the only one that is close enough in terms of speed is that Google, the black bar there, the Google Edge TPU on a specific neural network, the MobileNet V2. If you use the same chip on something else, it simply disappears. The only one that is fast on all four architecture is ours, and that is a testimony to the general usability of the chip. That is not easy to obtain, and that's why I want to underscore. On the left there, I have the radiation tests results. Essentially, they match the HPSC requirements. Actually, that 75 MeVs is actually wrong. That's my fault. It's a typo. It's 90 in our case. 90 MeVs, so it's even higher. Okay, we can go a little bit further. So that, as we mentioned before, space heritage is really important. You want to test these kind of solutions in space. As we mentioned, the spectrum, the type of radiation that you're getting in space, you cannot replicate on the ground. You can only emulate with some tricks. So you want to test this. We have essentially three opportunities. The first one we had was a suborbital flight on a higher altitude balloon. So this was supposed to be launched May last year. To this date, still not launched. We had so many different reasons for it. Whether even jamming test in white sand missile range, one day before launch, they said no, there's going to be this jamming test, so there will be no GPS. And others, I mean, just bad luck with that mission. And then we have an orbital flight coming in November this year for a 2-year mission on the NASA Odyssey. We are also in discussion for a constellation of satellites with NASA, SSDS, that will be launched in 3 different steps next year. Okay, so this is just to say we really care about this. It's very, very important for us. This is a common slide that we shared with Microchip, who are the guys that designed the HPSC. Why? Because we both are marketing our solution as the AI coprocessor of their host processor, of their main processor, the HPSC. We are the booster for that system in order to provide enough AI computing power. Essentially, you get the double of what they have with 1/10 of, actually 1/100th of the power consumption. You're just getting performance that you would need 2 of those chips essentially to achieve, and it's not even that. So tops, it's an indication, is not exactly the full indication. You need to see how you are going to use that computation amount. I can guarantee it's more than the double in terms of final throughput in terms of AI. Okay, software integration. I don't know if in the audience there is somebody that works on integrating coding AI solutions. This is a very important part. It's extremely simple to implement and integrate our solution in instruments, in a mission. So what we do, this is the software flow. What we need is essentially a pre-trained neural network in the most popular formats, which are called PyTorch, TensorFlow, Onyx. And a subsample of the data sets. So the data sets is the data you use to train the neural network. We use those 2 parts in order to compile, compress, and compile and optimize a neural network model for our hardware. Once you get that compiled model, you can move it to our development kit. And there, with the neural network running with our hardware, you can optimize all your actual application, right? Once you are happy with it, you can deploy that neural network on your platform equipped with our chip. The software will tell you if something doesn't work, why it doesn't work, and each layer you will get the time, the power, and the memory consumption of each layer so that you can optimize your model if you want to have the best performances out of that model. Okay, lastly, a very important point for us, we do have a development kit that is in the hand of a lot of groups at NASA. commercial space companies, and also not only space, of course, but also commercial space company. This is the Gen 1. Our chip is the one on the right in that picture. On the left, there is you can see another chip, big chip. That is an FPGA. Essentially, that chip acts as the interface to our chip. The performances of this are those of the Gen 1. So it's 2.4 tops, 60 milliwatt for our chip. It's very, very, very low power consumption. We provide, of course, the SDK to run and to try neural networks that the end user wants to try, and we also support the end user. So I would stop here at this point. If there is any questions, the next slides. I believe those were from.
I'm back on now.
Oh, okay, Michael. Maybe I don't know if you want to continue on this or.
I can continue.
Okay.
Sorry for the unintentional demonstration of why you want to do processing in SUTU as opposed to bandwidth limited, delay tolerant, which obviously this audio channel is not. So let's move on. And once again, thank you so much, Mirko. This is really a game-changing development, both in terms of the power efficiency and the radiation tolerance. I was with Mirko and Fernud at the 88-inch Berkeley cyclotron and absolutely amazed at the performance of their chip under bombardment with argon and krypton ions. It was actually spectacular, exceeding HPSC, and one of the HPSC radiation leads was with us. So really fantastic chip, and if you're interested, please do get a dev kit. So let's go on there and saying how can we get towards autonomous edge supercomputing? There's a couple ways to do this. One thing I want to highlight is that we are looking at a new interface standard from PCIe, which is more than half of the wattage on that Mentium chip, to being able to put everything onto a silicon wafer and greatly reduce the power and increase the throughput that you have. And so universal chiplet interface is coming on board, and it's going to become a very widely used standard in about 2028. So this will help with the integration of multiple processors and provide some what I think will exceed 100 AIML teraops per watt. Let's go on to the next slide. Now, I do want to go back to the origins of neuromorphic computing and look at what are the differences between what has already been achieved in neural net computing electronically and the human brain. So one option that we're looking at is original equipment manufacture. That is a biological neuron. And the way this actually would work is that there is a platform called Cortical Labs 1, and it allows you to put in not just a brain organoid, which consists of neurons and astrocytes, and then they're connected with microelectrodes, but actually a system of those. So you can actually get something which would be corresponding to the differentiated neurons that you see in a human brain. And what you do here is you take the blood of a donor, like an astronaut. Perhaps you're trying to mirror the astronaut's brain so that you can develop precision mitigations to radiation. And you push it back into a generic stem cell, then differentiate it into neuron astrocytes, and then be able to grow those and then put them onto microelectrodes. And this way you can actually do computing. Here's a reference in the Neuroscientist about how you do the harnessing of brain cells to being able to do computing in situ. Next slide, please. Now, the neurons actually have a greater dynamic behavior than the kinds of electronic circuitry, such as that array of memristors that was shown early on. 1955, this is a model was using ordinary differential equations of the Hodgkin-Huxley model for neurons. It consists of three different kinds of ions, sodium, calcium, and potassium. And their interactions as they go across the neural membrane leads to what's called a neural spike. And the modeling for this, done back in 1955, is able to give you not just the behavior of a single spike, but if we go on to the next slide, please, very rich dynamic behavior. So with a very few number of neurons, you can get oscillatory motion, such as a fish swimming, or the way that you do writing is you are essentially modulating oscillations with your hands. So 1999, Professor Asafekic at MIT, he showed that there was 20 different dynamic modes for neurons that give you a lot of behavior, more so than what we have electronically. So that points to the fact that maybe we're not at the point, even with everyone predicting in 5 years that we will have general artificial intelligence, of being able to mirror the performance of biological neurons with electronic ones. And in fact, there's a reference that you can follow if you look at B.J. Kagan, references 2023 to 2025, you'll see that biological neurons actually do better training for challenges like the MNIS, which is identification of digits, or being able to play games like the game of Pong, than electronic versions based upon the tensor processing. So this provides some insight that maybe we have more work to do in terms of neuromorphic computing in order to identify what human and animal brains are able to do as compared to what is there electronically. So I think this is very exciting. And then we can go to the summary of for future space computing. Going back to the beginning, space qualification requires deliberate engineering testing to meet mission profiles for radiation temperature and other environmental conditions. Yes, you can put up experimental processors, assuming your flight board will allow you to do that. As you move off to something where you have long-lived missions that are in high radiation environments, you have to go up to a PDR. You're going to be challenged, and you need to be able to show to those review boards that you are meeting the environmental challenges. Second one is that heterogeneous capability, such as neural cores, have already been achieving for upcoming generation of high-performance space computing, as you saw with Mentium. But if you pick up your cell phone consists of chips, systems on chips that have both CPU cores, GPU cores, and neural cores. And the neural cores are the ones that give you another order of magnitude of performance as compared to 2015, 2016 cell phones. That's why I can actually get transcriptions off my cell phones that are working real time without being connected to the internet. So neural cores as they exist now electronically are able to greatly accelerate the kinds of AML. And then when you go to space qualified neuromorphic processors, of which Mentium is probably the first out of the box, it can greatly accelerate AIML algorithms in space, especially with low power. And finally, going back to the origins of neuromorphic computing, neuroscience is progressing rapidly. We're beginning to understand the mechanisms, the rich behavior of neurons, and it could end up being that the biological option might prove to be superior to electronics for tasks beyond inference, such as training new models. I think we'll learn a great deal in the next 5 to 20 years about that. Maybe we can mirror that electronically, or it could be that the biological neural membranes really provide something that is above and beyond what can be achieved electronically. So neuromorphic computing has already achieved a great deal. It's already pervasive in terrestrial computing, on your cell phones, on your desktop. With progress such as the Mentium DVMR, we are going to be able to use that in space. And then perhaps in 5 years, we don't have general artificial intelligence. It could be because we don't have the equivalent of the biological neurons, and we should continue to learn from neuroscience. Thank you so much, and if we have time, I know we're a little bit over, I'm certainly able to answer questions, as would Mirko.
Yeah, thank you so much, Dr. Prezioso, Dr. Lowry. That was outstanding. I know there's been a lot of excitement around neuromorphic computing, and following both your work for some time now, this was just a big honor. And I'm certain both the AI/ML from the software and hardware side, maybe from James to Pedro to Lauren in the audience might want to come chat with you now. Also, I'm sure like Wendy or other the brain neuroscientist people, I think would be keen. So, but we are going to move on to our European Space Agency presenters now. So, thanks so much, Dr. Lowry and Dr. Prezioso. But if you can stick around, maybe in the lobby, you guys can have some Q&A with some of the participants. So that was really exciting. So I love the breadth of the presentations we've had so far. It really touches on all aspects of what the AWG wants to strive to support. So cheers. Thank you.
Thank you.
Ryan, thank you for the opportunity.
Just give me a moment. Rodrigo, fantastic. All right, so going to unspotlight, and then Mirko unspotlight. Okay, let me go full screen. Okay, so we've got Rodrigo Almeida. Let me see, is Cesar here? I don't see him, but I think, there's Claudio. Perfect. So I've Claudio and Rodrigo, and let me get you Michael up to Michael Olufemi. Maybe you three want to turn on your Claudio, thank you. Hi. And then Michael, is your camera working? Looks like maybe. Might have to enable your browser, but we have time. I'm going to share the screen now, and I'm very honored to introduce Rodrigo Cucina de Almeida from the European Space Agency, and he's going to be giving a brief introduction to about this session. So, welcome, Dr. Cucina de Almeida.
Thank you, Ryan. Thanks a lot. I'm happy to be here. I'm going to give you just a brief overview of the space omics, but also a collaboration that's been going on between ESA and NASA Genelab for now, but you'll be in the future. We hope to enlarge this. So just for everybody to know that ESA Human Robotic Exploration has a specific data archive, which is basically a repository of all our data that goes to different platforms, including the ISS, but also parabolic flights and other type of platforms, including historical platforms. So I put the address here, hydraisac.esa.int or hida.isac.esa.int, and you can see that. But what was the idea here? We saw that there was a lot of omics data. We want to start this collaboration with omics data. Then we got in touch with the folks from NASA Genelab, and that's the start of collaboration with them. The next slide. So the idea is to have all the omics data also using the pipelines from NASA Genelab. So in that way, our scientists and the community can benefit from this, And we have all this data hanging around, and we're not going to reinvent the wheel. And NASA Genelab has already all the pipelines and all settled. So this is ongoing now. I'm very happy that this probably will be by the end of this year. We'll make available to the whole community. There is some technical issues, but it's ongoing. So the idea please next one. So to encourage the scientists to explore and to how we're gonna engage with the community, we have this idea last year to have a Space Omics hackathon. So this is that you're gonna see the two winners, the talks. But this was a hackathon dedicated for tackle some of the challenge that we thought, together with the experts, including members of NASA Genelab. Next, please. So we have proposed for the teams for the hackathon 6 challenging from reproducibility and open science, which it's a big topic, but also space multiomics, but even go to planetary protection. Next, please, Ryan. So what we have there, just to give you an overview, we have 23 proposals. We have we because of manpower and also we couldn't make it. We have to select some of them. Of course, we wanted to do all of it, but we select 10 from these 23. But we saw there is a lot of potential in the other ones, but we have to put some filtering. Unfortunately, cannot see in this slide here. Apology for this because in the PDF I think it screwed up a little bit. But what you see here is the distribution of the topics that the teams chose. And then here, what the model of this data set. So also indirectly, we have much more microorganism than any other type of organism. Next, please, Ryan. So, and also the distribution to see of the team members. We have from the Netherlands to the US and even Japan. So it's a very, very large distribution, but it's just good to see that, this is worldwide and we can embrace all type of the whole community. Next, please. And then, as the winners, just to thank the winners, and we visit the European Space Center, but we also had the winners in doing a internship with NASA, and also we will sponsor the publication of this. And next, please. So what we learned, so lesson learned from this initiative, that this reflected the relevance also for our community, for the open science. We successfully engaged, even though it's a totally online, but from this community we know that this is possible and we can do much more than we thought. And I think the hackathon also brought new ideas and methods, the machine learning and AI for the already published data, which is what we've seen here also in the AWG. And the last one, we also saw a good collaboration between the mentors and the students, which will lead to is sponsored by ESA, open access publication, and also you continue this collaboration between a group and a well-experienced scientist. And with that, I would like to thank you. And if you have any questions, I left my email here, and that's it. I'll leave the floor for you, Claudio.
Thank you so much, Rodrigo. It's so good to hear that overview of the collaboration and the data coming up. And the Claudio, are you going to go first, or is Michael?
I don't care.
Let's have you go first, then, Claudio. I have it open so I can get that first. And then Michael, it looks like when you're ready, maybe test out your camera too. But I also have your deck all set as well. So welcome, Claudio. Cheers.
Yeah, so thanks for the introduction. My name is Claudio, and I will be presenting to you briefly the project that we did for the hackathon that took place last year. And no, not yet, sorry.
Sorry.
So the main idea of the project was to, we have all of these experiments, but we know that the experiments that take place in space, they have a lot of covariates, so a lot of different metadata that are all mashed together in what you're measuring. So if you're measuring, for example, transcriptomics, you know that the transcriptome that you're measuring is sort of the effect of all of these different covariates, like radiation, microgravity, the age of the mice, or the strain of the mice, or the species of the plant, whatever. So our idea was to, can we use the already published data to kind of like learn more from this sort of data? And now you can go to the next one. Yeah, so I won't go over the complex math or anything, but we trained an AI model that is based on variational inference. And variational inference, what it assumes is that your input data, can be explained by a combination of latent factors. And these latent factors are sort of like a representation of your data, And what variational inference is going to do is that it's going to learn a distribution over these latent factors, such that we can take this distribution that we learned with our AI model, and then we can take new samples from this distribution, and that's the generative part of the model. And that's the variational inference part of the model. And then you can go to the next one. And then what is disentanglement exactly? So that's the title of the talk. So traditionally, computer science algorithms have tried to take your input data and have tried to classify, for example. And in order to do this, what they do is that they learn a lower dimensional representation, so these latent factors. And then on top of that, they build a classification model that is able to predict the different inputs that you have. So to illustrate this entanglement, I chose one of these examples that is based on computer science, but I think it's good for illustration. And that is that, so traditionally we would have learned a representation, so a combination of latent factors that would be sort of a black box. We don't know what this representation means. And what disentanglement tries to do is that we try to train, no, before, sorry, not yet. So what we try to do is that each of the latent factors, we want each of them to learn something from your input data. So for example, the first one might learn something about the angle of the digit in this case, the width of the digit, or the thickness, or you can think of any other descriptors. So you can go to the next one. You can skip this one. You can go to the next one, actually. Yeah. So then we thought, okay, can we apply this concept to spaceflight? Why would that be useful? So our selling point is that spaceflight is going to become only longer, and then astronauts are going to become exposed to radiation levels that we haven't seen in experiments. So then if we are able to disentangle this radiation effect from the experiments that we already see, then we would be able to predict the effect of radiation that we have not yet seen to the transcriptome in this case by training an AI model. You can go to the next one. So we took this data set specifically that took mice, and they had a control group that stayed on Earth, and a group that was sent to space, and the mice were exposed, of course, to different radiation levels and microgravity levels. They specifically measured the hippocampus, and they did spatial transcriptomics in this, but we didn't really care about the spatial or the organ itself. We just wanted some transcriptomics data that had different radiation levels to be able to disentangle this effect. How we do this is by training an AI model. I won't go into the details of this, but we basically take our input data, our gene by spots matrix, and then we train a model that is able to sort of disentangle this radiation effect. And you can go into the next one. And then once we have understood what is the effect of radiation on the transcriptome, what we can do is that we can actually tell the model, okay, can you try to predict the effect at different radiation levels? And that's exactly what we did. So we took I think it was 5 different radiation levels that were measured in the experiment, and we interpolated the values. So we have a continuum of radiation levels, and then we predicted the gene expression at each of these points. And then you can get some sort of curves that you see here on the slide. And then by fitting some functions to these curves, we can group the genes based on what genes increase in expression as radiation levels increase, and what genes decrease in expression as radiation levels decrease. I won't go into the biology because of time, but we found some genes that are associated with radiation levels. You can go to the next one, and last. So sort of like future perspectives of this model, of course, it's just a pitch idea. It's not anything definitive, of course. But I think it would be useful if we know what genes are affected by radiation specifically. We could, for example, map those genes to diseases that are happening on Earth right now, for example, and then there might be some drugs that we can repurpose for this already. Then, of course, we would like to extend the model to different tissues, different covariates, more data points. That would only improve the model and make it more robust. And then, of course, the data had a spatial component to it, but we completely ignored it. I think it would be really interesting to know what is the effect of radiation on the distribution of the different cell types, for example, on the tissue. And with that, I'll finish. I'll be around. You can ask any questions or we can talk.
Yeah, I was just about to say, yeah, Claudio, that's outstanding. I really hope some of the Brain AWG members and AIML, it's a very robust community, Claudio. So I think if you're looking for, taking that collaboration to the next stage, I'm sure there's a lot of interest in that. So next is Michael. I'm just pulling up your slides. Michael, welcome. Full screen. And all right. Fantastic. Yeah, welcome, Michael. Hello.
Yeah, thank you. Can you hear me?
Yes. You're coming across totally clear.
Yeah. So yeah, my name is Michael. I'm presenting on behalf of my team at Bio. We are part of the hackathon for last year and we came first place. So our project was to model tissue-specific omics to find a way to predict space conditions using deep learning. So those are my team members, and they are not here. I'm presenting their behalf. So the problem, or like the problem in disguise, is the rich dataset from space biology. They are fragmented because they're from different missions, so they're different tissues, different species, and sometimes they're on different platforms. So all we wanted to do was to find a way so that we can integrate all of this together and find a way to get meaningful insights by predicting, interpreting, and finding a way to guide future experiments so they can simulate outcomes in silico before expensive space flight. So to do this, next slide, we try to use this different mission data sets. We had about this large amount of data sets spread across different tissues and sexes and different tissue samples. So we had about over 800. And next slide, this heterogeneity in this particular rich data set is what is the feature that we are trying to exploit in our model. So we want to use a compositional variation autoencoder to model this multi-omics. So what we are going to have is this high-dimensional multi-omics data that we compact into a latent space. From the latent space, then we can find a way in latent space that encodes all these different covariates, the tissue, the exposure, and the different sexes that you have. Then we can then decode back to see, can we get this same gene expression levels and methylation profiles even for unseen condition. And compositional here is that we're able to factor the latent space into the parts that correspond to covariates. So the tissue species, they have their own learnable components that can that we now compose into a final state. Next slide, thank you. So in this slide, you can see that our model this is the training of our training dynamics. You can see that there's no late epoch optic, and there's a low final error, which supports that there's a trustworthy latent for the downstream interpretation and simulation. And a lower MSC score shows that we have a better reconstruction and prediction rate. Next slide. So we were able to project this latent appendice, and we're able to separate the different samples from our model, the ones that were exposed to ground control and space flight, so we can effectively distinguish between from our data sets this basal and space flight. Next slide. We are also, because of this heterogeneity, we are able to effectively differentiate the different sex differences, and this is because there's a relative treatment per gender. So the model reflects that this little separation of the sex in latent space. Also, we're able to capture tissue specificity in our model. So you're able to predict for almost all the tissues that we have, the different embeddings that they have. And beyond clustering, we're able to rank genes. Next slide. We're able to rank genes by how strongly the model attributes expression. So we have like this round score of 0.6 that shows that there's a separation of each of the responsive genes. And this is useful for pathway analysis and hypothesis generation. And we can use this we can also predict this in a tissue-specific manner to show how a particular gene is going to respond when it is being exposed to space flight. Next slide. So this is what we are able to do. We've been working on this for close to a year now, so we are trying to increase the modality of the input data. At first, we thought we can find peer data for the different multi-omics data, because we're really interested in both epigenetic and gene expression data sets. But no, we had challenges about this short data that we could not pair together for both methylation and gene expression. And so that's next slide. I think that's our last slide. So yeah, so this is what we have. We have this deep learning approach that we can use to predict different multiomics and experimental covariates. And we acknowledge that we are limited by our input data size and computational power to train our model with more data sets. And with that, I'd like to thank everybody for listening and ESA for helping with the hackathon. Yeah, thank you.
Thank you so much, Michael. Just a side note, Michael also had completed an internship with his colleagues this past summer with the OSDR team, where they took on a whole other different challenges and helped create a simulated plant agent, an AI agent for plant health support, integrating a lot of different, from documentation to data to multimodal data as well. So keep an eye out for future talks on that project. And yeah, hopefully Michael, you can stick around maybe a little bit, and others can talk to you about your ESA project. So let's take a 10-minute break, and we'll come back and talk about the new AWGs and opportunities. And so we'll return in 10 minutes, so 10:20 Pacific time. So thank you, everybody. We'll be back here in 10 minutes.
Hello, everybody.
First.
Do you want to introduce yourself, Nilafar, first?
Sure. Hi, my name is Niloufar Ali, and I lead the PD part of the Brain AWG, but then we are starting this Brain AWG with Wendy, and I think Wendy will take you to what our group is about, and then I'll jump in later.
Thank you. So I'm Wendy McNerney. I am very excited to announce the newly formed Brain AWG, which I am co-leading with Dr. Nilafar Ali. So this is a question that we're trying to tackle from a neurological health perspective. So neurodegenerative diseases is something that nobody should ever have to experience, especially within the face of service to their country or as having work-related contributions to risk of this disease. And we're pretty sure that going into space and spaceflight does increase this risk, just given all of the exposures and stressors associated with spaceflight. And so this group would like to tackle this question, and specifically as we learn more about conditions that increase neurological disease, we can start developing effective countermeasures towards neurodegeneration to help keep our astronauts safe and to keep people on the ground safe as well, to reduce risk. So we tackle this by using advanced multi-omic and data analysis approaches. As you can see listed here, we have a lot of subgroups, all looking at this question from different perspectives, from Parkinson's disease and the Alzheimer's side of things, which I'm leading. We also have stroke, TBI, and looking more specifically at certain pathways in more general. So we have a pretty exciting approach. And now I'm going to turn this over to Dr. Nilafar Ali to explain some of the results that she has with her research.
Thank you, Wendy. So as Wendy mentioned, we have a lot of subgroup and interest group here. So it started like with the more mature group that we have is the PD subgroup because it has been going on for a year, almost more than a year now. What we essentially take on as an approach for most of all of this group is first we do scanning of whatever disease we are looking at. Say, for example, here we have projected the PD subgroup where I like analyzed all this data, like first curate data from PD patients. So, for example, from Michael J. Fox Foundation, PPMI, from published literature, from GEO and CBI, wherever you can get the most large amount of data from PD patient, and then compare it with whatever similar groups of tissues we have with the space flight condition. Say, for example, we have the data set from the iPSC dopaminergic neurons or organoids, and then we are comparing with the space flown organoids of the same type. Then blood from the Parkinson's disease patient, and then we're comparing it with the blood from spaceflight from the astronauts, whatever data is available in the OSDR. Now we are looking at the common markers like the pathway trends, and then we kind of generate our custom pathway for this individual group of diseases, neurological diseases, and come across like what are these things, what are the things which are common across this two different conditions, which shows very uncannily some identical results. So we are also looking for countermeasures. Here is one of the some data that we have published. We also are going to have more publication in the future. The deliverables are, yeah, we have this presentation, papers, and publications, and also like we are looking for collaboration and writing grants in the future for experiments. We are a very highly collaborative group. We have inter AWG collaborations going on, even newly started with the plant AWG, where we are looking at the lunar regolith and neurological influence. So a very cool set of projects that we are having, and we are always looking forward for new ideas and collaborations.
And we have a couple of presentations today coming from our subgroup, so please tune into those. They're going to be very exciting.
Hi, welcome everyone. Thank you. I'm new to the AWG family and really excited to announce the Planetary Protection AWG with Stefan Green and Alex here as well. So our primary mission is support the goals of the planetary protection by making and developing unified and international standards for bioburden assessment. So we're really looking at a combination of standardizations of methodology, including DNA extractions, recalcitrant microorganisms, bioinformatics, pipelines, and additional tools and protocols complementary to the standard assay. And secondary goals are really these ultra-low biomass protocols, contamination workflows, both experimental and in silico, and working with commercial entities to develop DNA-free reagents to really try to mitigate the contamination issue with low biomass samples. My name is Haley Sapers, and I'm an assistant professor at Northern Arizona University in the Planetary and Astronomy Department and also in Biological Sciences. I'm mostly working on high performance DNA extraction protocols, maximizing recovered observed diversity, as well as looking at microbial standards and different DNA-free reagents for these molecular biology workflows. So I'm going to turn it over to my co-chairs to introduce themselves and some of their projects as well. And again, we really just want to encourage people to join the group. We'll be opening up membership in January.
Great. Thanks so much, Haley. Brian, thanks for going one more. Hi, everybody. My name is Stephen Green. I'm a microbiologist and microbial ecologist working at Rush University in Chicago. And together with Haley and Alex, I'm a co-chair of this new AWG. So I have fairly long pedigree with NASA-related microbial work. I did a postdoc at Ames Research Center about 20 years ago, looking at hypersaline microbial mats, and now have moved on to more clean room style microbial community analyses. One of the areas that I've been working on this particular AWG is looking at how low can we go in terms of low biomass for metagenome sequencing, which is we're hoping will be a complement to the standard assay using cultivation. And so we have multiple reasons why we'd like to be able to add cultivation independent methods to our assessment of bioburden. But one is that the standard assay only looks for aerobic spore forming microorganisms, and we'd like to be able to detect a broader range of organisms that could also be of planetary protection concern. So one of the areas that I've been working on is again, ultra low biomass and whether we should spike in DNA, carrier DNA into our samples. We're actually about to submit the first publication, or the first manuscript from that tomorrow. Keep telling myself I'll do it tomorrow. And showing that actually that carrier DNA is not particularly useful for our applications. So happy to turn it over to Alex now.
Yeah, hi, I'm Alex Mahnert. I'm located at the Medical University in Graz in Austria. So I'm the European ingredient in this co-chair group for our PP AWG. So most of my projects are supported by the European Space Agency, but I was also an intern at NASA Jet Propulsion Laboratory. So my background is more like a dry lab computational background. So the projects we're currently working on are, for instance, metagenomics projects where we try to understand which variables, which data points can be implemented in probabilistic approaches so that we can do a risk estimate. So this is kind of interesting for the implementation. So we need to break down this very complex data sets to for mission teams that are not really from the field of metagenomics, for instance, so that they can take action and make the decision if the mission can fly or not fly. So what we are working on are also reference databases. So not only NASA Gene Labs, but also the German collection of microorganisms. There is a so-called ESA strain collection for space microbes. And this reference database will be helpful that we can also then do real-time sequencing, for instance, with adaptive sampling to this database. And we are also trying to make standards in metagenomic workflows, for instance, And it would be also interested to reach out to the community then, that they can either test our workflows or give recommendations, or maybe also show us a better workflow for the analysis.
Sure. Yeah, we're definitely welcoming people who are interested in joining, really looking for a wide range of expertise. This is a really interdisciplinary group. Some of the areas that we've highlighted are expertise in nucleic acid extraction, metagenomics, bioinformatics, risk predictions, engineering control, clean room setup, low biomass standards, DNA-free reagents, low binding plastics. These are all some of the areas that we're interested in. We have a combination of lab and field expertise. Myself, I do a lot of work in the field up in the high Arctic, and also blood from a stone, so extracting nucleic acids from rocks. We connected with meteorite folks and some of their pipelines for carbon contamination, ashing. So really a wide range of expertise and a really interdisciplinary group. We're currently in the restricted membership phase and will be fully open in January 2026. So please get in contact. We're really excited to extend the invitation.
And Ryan, can I add just one last thing? Haley, great job. Just one other mention that we're also interested in partnering with commercial entities, since we, ultimately will want off-the-shelf solutions, to provide to any space agency. So this is one where we really would like to work hand in hand with our commercial partners, to again drive the microbial biomass in reagents and consumables down as low as possible.
Yeah, can you hear me now? Okay, great. Okay, I'm Jessica Lee. I'm a research scientist at NASA Ames Research Center, and I'm here to introduce the hardware AWG, which is really quite brand new. Next slide, please. Okay. So why are we starting a hardware AWG now? It's because OSDR is about to release a new repository. As part of OSDR, there will be a database called SPLASH, which is the Space Physical and Life Sciences Archive of Scientific Hardware. That is going to be a database specifically about the experimental hardware we use to do biological and physical sciences work in space, so that you can go and find information about all of those instruments. I won't go into detail on that because Kylie Brumma, an intern from our project, is going to present this afternoon Pacific time in the second science session today. So go and hear her talk to find out all about the database, what's going to be in it, how it's structured, and all of that. The AWG is our way of getting community input for this database. The AWG is going to focus on improving and using this hardware database. That said, if you're interested in hardware related questions and don't really want to work with Splash, you are welcome to bring those project ideas also to this hardware AWG. Our main goals for this AWG are to encourage exchange and standardization of information relating to spaceflight hardware, to ensure that the database SPLASH is accurate, complete, and up-to-date and useful for you, the community who might use it, and to foster this community that might be interested in doing spaceflight experiments and developing new instrumentation. And so then my final slide has information on, oh, I'm sorry, you can just click all the way to the end. I didn't mean to have all this, the animations. So we meet monthly on the fourth Thursday of the month. Up until now, we've had just a couple of sort of starter meetings to kind of test the waters and get an idea of what our scope should be. But going forward from a month from now, we will be having monthly meetings. And I'm presenting here some ideas for some subgroup projects that we would like to get started. The first listed is the manuscript. So Kylie and I and a few others are currently working on a manuscript to describe the database to the community to publish in December when we're planning to release this new database as part of OSDR. There is a potential to do more with it, to do data mining as you all are used to doing, and to incorporate some of that into this manuscript or maybe into a future manuscript. So if you're interested in data mining on the spaceflight hardware that are out there, maybe finding out what capabilities are lacking, or drawing other conclusions around spaceflight hardware, that would be the topic for you. We also have adopt an instrument. So if you are a user or developer of a particular instrument or a family of instruments and you want to adopt it essentially in Splash and make sure that everything in the database is complete and accurate and you're the best POC for that, then please join us to do that. And we can welcome you to the team to help, with that curation of data. And then finally, data standards. And this is something that we have not spent any time on yet for Splash, but we do want to make sure that the data about the hardware, like the technical specs around each instrument, the information we have about its potential uses and what other instruments it interfaces with, and et cetera, that all of those are in a format that is maximally usable. And so we want help from the community to understand how best to do that. So if you're interested in joining, please do. We are open and welcome to anyone joining, and just sort of find us on the AWG space or just contact me and we can have a chat. Thank you.
Can you hear me? Is there any way for me to share my screen? I can do that. Okay. Yeah, thank you, Ryan, for the invite and letting us talk about physical science informatics. I'm the lead scientist for PSI, and I'm a material scientist by training. But enough about me. I would like to introduce the rest of the team. We have a very small team at PSI. Sean is our manager, and Cassie, of course, is the lead coordinator. Karen came back from retirement to help us, so some of us are even working part-time. Pranav out of Ames is our curator and everything AI. And then, of course, last but not the least, Matthias, he's the lead developer, does all the background coding and everything. So that's the team pretty much. Next slide, please. So what is this PSI? Physical Science Informatics is basically an open science data repository. It goes back to the shuttle days, and it encompasses 6 different areas of science, and I'll talk to you about that in a second. And it's not just a data repository, but for the last few years, we have been supporting NASA BPS, Rose's call for proposal, where you're expected to use the PSI data to submit your proposal. So use existing data to build new insights, and that way we accelerate the collaboration and also the timeline from research to the product, right? So that people can publish papers and collaborate on new proposals. So currently we are hosting about 117 investigations in 6 different areas, and that's about approximately 34 terabytes of data. And we are continuously adding more data almost on a weekly basis. Next slide, please. So when you go into the data repository, what you are going to see on the left-hand side, the top, we have 6 different areas: material science, combustion science, fluid physics, fundamental physics, soft matter, um, and even biophysics. Something you guys might be interested in has data investigations on protein crystal growth, amyloid fibrolysis, and things like that. Um, Once you click on any investigation, and you can search for investigation based on the areas of science, or whether you're looking for ground-based data, microgravity data, that is shuttle or space station, or reduced gravity, parabolic flights and such. And once you click on an investigation, you will see that the data is organized by the description, that is the metadata. We have an experimental table telling you about the samples and the parameters used, about the hardware, like we just heard in the last presentation, and of course, all the raw analyzed data. Most importantly, the publications and the reports and even dissertations. So that's the content for each of these investigations. Next slide. So what are the most recent updates? So PSI went through a pretty big update in the last year. Before that, it was just a hodgepodge of data, very difficult to navigate. But now we have gone to the cloud, AWS cloud, and all the investigations, they have a similar layout like I showed in the previous slide, and the file structure. We have a DOI for each investigation because that's something the PIs wanted, because when they publish, they need a DOI to point back to their raw data. We have added a submission portal so that I don't have to do most of the hand holding. The PIs can actually go into the submission portal and submit their own data. And now coming to the AWG, we actually did not have any AWGs for any of those science areas. So we are doing a humble start with combustion science. We are going to kick it off on the 7th of October. Yeah, thank you. And I learned a lot from Ryan how to start the AWG. So thank you. I was hoping maybe we would get 5 people to start with. We got 15. These are all people from academia and also industry. So I love that mix. And hopefully we'll also combine the European database, that is the HREDA, because many of their PIs and our PIs work together. So we are looking forward to that. And hopefully this will be a success, and then we going to start working on the other areas. Now, if looking at the picture at the bottom left, this is the new site. Actually, NASA is consolidating all these databases into one website, and so that's our PSI website. So the 3 main buttons are the data repository. Anybody can go in there. It's totally open. The submission portal is just for the principal investigators and for people like myself. And then we have a tool, an analytical tool, which we are testing out. It was a tool developed using NASA PSI grant. So we are making it available for the general public. Anybody can create an account, takes 2 minutes to create an account, and then you can get in there and use it for analysis. And actually we had a presentation about that in the joint user group meeting just a few weeks back. So I encourage you to go try it out, see if you find it useful, give us feedback, please. What else? We have added 3.5 terabytes of new data in 2025, and like I said, we are continuously adding more. And very soon we are going to have a AI assistant in within PSI, and we need also help on that from the AWGs, like what they want to see the assistant do. Right now, we should have it implemented within a few weeks. I like some of the features already. They can give you a summary of the investigation. They can compare different investigations, like how science has progressed from the shuttle days to the ISS days. Those are the recent updates. And if you have any questions, interests, please reach out to me. Thank you. I hope so too. Yeah. Thank you. Bye.
Can you all hear me okay? Yeah, excellent. Okay, hi everybody. I'm Catherine. I'm one of the co-leads of the biofilm subgroup that's associated with the microbes analysis working group. I co-lead the group with my colleague VK. So I'm based at the University of Glasgow and VK is based at Amity University in Mumbai. And I'm delighted to be able to talk to you about the projects that we have been working on this past year. Next slide, please. Oh, back one, please. Oh no, yes, right, you're right. Sorry, that one's the right one. Sorry. Yeah, so I appreciate that like we've got a broad range of backgrounds in the room. So I thought I'd talk about give a brief sort of background on biofilms. So the main lifestyle of microbes on Earth is biofilm. Biofilms are mixed species communities of microbes which contain bacteria, fungi, archaea, and viruses. And these are encased in a matrix of extracellular polymeric substances that they have produced themselves. And this matrix protects them from the external environment. Biofilms are very heterogeneous. Their structures dependent on the microbial species that we find within them and the environmental conditions that they grow in, and this makes them very highly adaptable structures. And this is reflected in the fact that they're found in every ecological niche and they're essential in the maintenance of life on Earth. So they're involved in a range of different biogeochemical processes. They're involved in oxygen generation and carbon dioxide processing, and they're key in maintenance of organism health. However, their persistence and adaptability means that they pose serious problems in areas such as the built environment and in healthcare. And when we extrapolate this to spaceflight, fundamental roles of biofilm in the support of life on Earth kind of like opens up possibilities of biofilm-derived applications for this life support and space exploration. However, there's also a threat from biofilms through biofouling of the built environment, like for instance in the water supply, biofilm-related infections arising from compromised crew physiology, and the contamination of fresh plants by both plant and human pathogens. So really, to support crewed space exploration, we really need to understand how biofilms behave in the spaceflight environment. And this means we have to kind of understand how biofilms themselves with the community reacts, how the biofilm interacts with the host, and how biofilms interact with craft microenvironments. Okay, so next slide, please. So the aim of the biofilm subgroup is to apply the OSDR data to understand the biofilm challenges in spaceflight and provide novel insight into biofilm behaviour. We have 3 projects that are running now in various stages of progress. The first one, where the forward-looking perspective, focuses on the positive aspects of biofilms and identifies our current understandings and knowledge gaps. And that's the project that I'm going to talk about for the rest of the presentation. The other project we've got running just now that started a few months ago is a manuscript looking at methods to study space biofilms. So we're collating and detailing methodologies in growth, quantification, omics profiling and imaging, which have been used in space-flown and ground-based studies to basically kind of like capture all that information. And that's the project that VK is leading on. And our third project is the analysis of reactive oxygen species in microbial and biofilm OSDR datasets. And we're in the initial stages of identifying datasets for this project. So this is quite an early project. Okay, so next slide, please. Okay, so the prospective review that we have it's currently under review in MPG Biofilms and Microbiomes is entitled Biofilms from Cradle of Life to Life Support. So we kind of undertook this as a scoping exercise to identify knowledge gaps and highlight potential research questions. So we focused on literature in 3 main areas. So how biofilms on Earth support life, how spaceflight impacts biofilm formation and the biofilm host dialogue, and how we can leverage space to drive forward biofilm discovery and innovation. Next slide, please. So we began with the evidence of biofilm emergence early in life's history and their function as precursors of multicellularity. So conditions on early Earth may have promoted accumulation of macromolecules, which led to partitioning, compartmentalization, and the rise of protocells, which then developed into what we now recognise as biofilm. In the geological record, we see early biofilms as stromatolites, where sediment has become trapped in the extracellular polymeric substances that are produced by early microbes, and as fossilised fungal-like structures. We still see stromatolites today formed by cyanobacteria, which are capable of survival in conditions that are analogous to early Earth. So cooperative, I'm not finished yet. That's okay. Cooperative cellular behaviour arose in biofilm as a lifestyle benefit over that of planktonic cells, and most likely primed the emergence of multicellular life, considering like when we look at the benefits of biofilm over the lifestyle of single cells, because we can see similar function to that of tissue in terms of like resource management, the coordination of cell behavior and cellular differentiation. And then as more complex life evolved, biofilm became a facilitator to that life, so leading to today where biofilm is central to life as we know it. Now, next slide, please. Yeah. So we looked at the impact of space stressors on biofilm. So biofilms support health in a variety of different ways. So broadly speaking, these ways are very similar between humans and plants, where we have biofilms training and modulating immune responses, protecting against pathogen evasion, and providing nutrients to the host. Our review highlighted a variety of observed changes and adaptations in biofilm behaviour of both bacteria and fungi in spaceflown studies and ground-based analogues. And these changes varied depending on the species tested and the study parameters. So, we found that there was changes to quorum sensing, adhesion, biofilm architecture, antimicrobial resistance, and modulation of that extracellular polymeric substances. When we turn to host microbiome interactions in spaceflight, our understanding is very poor. So in humans and mice, causal inference from microbiome alterations and changes to the biofilm gut interaction may influence lipid metabolism, bile acid modification, and reduce capacity for defense against pathogen invasion. In terms of plants, there is evidence of biofilm support in microgravity-induced growth perturbation. However, there's no literature on the influence of or role of biofilm in other spaceflight-induced changes. From our literature searches on the impact of spaceflight stressors on biofilm, we found two prominent gaps in knowledge. The first being the lack of study of inter-kingdom microbial biofilms. So considering microbes live in these big multi-species communities containing bacteria, fungi, archaea and viruses in nature, this is a huge piece of the puzzle that we're missing. The second knowledge gap we've identified surrounds the intricacies of the biofilm host dialogue and the degree to which that's impacted in spaceflight. Since biofilm host interactions are fundamentally important for general health and well-being of organisms, this profound lack of information will impact our ability to provide effective countermeasures against spaceflight-induced physiological change in both crew and plants. Next slide, please. So, moving on from the impact of spaceflight on biofilm, there's significant opportunity from space-based research to drive future biofilm innovation due to the unique environment of spaceflight. So in addition, there's a host of new methodologies which can help us gain greater insights into biofilm behaviour. Advanced imaging systems, spatial transcriptomics, and multi-omic integration can capture signalling system dynamics, metabolic processes, and biofilm architecture in space, analogue, and low Earth orbit environments. AI and predictive modelling can utilise this data to drive forward image processing, biofilm host interaction prediction, feedback modelling, and design programmable biofilm for specific tasks. Our review also identified current biofilm-based technologies that we can build on to drive forward new innovation. We can leverage known signalling systems to encourage immune clearance or augmentation of antimicrobial treatments, and utilise biofilms and targeting systems for vaccines and drugs. Biofilm host communication can also be used as biosensors to activate drug or nutrient delivery responses, with probiotic organisms acting as drug delivery systems. Engineered plant-associated biofilms could also enhance crop growth, resilience and yield, producing nutrient-enriched fresh food and plant-derived medical provisions, highlighting the many ways that space provides an unprecedented opportunity to explore biofilm as a functional structure in its own right, as a component of healthy organisms, and as a biological technology. Next slide, please. So overall, our review has identified 4 research priorities for biofilm-enabled space exploration. So these are, there's a need to profile interkingdom assembly as beyond our multi-species prokaryote biofilms. We also need to understand the intricate functional dialogue of biofilm host associations. We need to kind of focus on technologies that will allow us to engineer biofilms to support life in space, and then translate all these kind of biofilm insights from space to enhance life on Earth. So the data produced through priorities 1 and 2 would drive 3 and 4 through AI and ML research. Modelling of biofilm formation and growth dynamics can then be applied to minimise health risk and promote health and well-being of both crewing crops and spaceflight, and then be translated to the current existential challenges that we face on Earth, including antimicrobial resistance and climate change. And in our review, we conclude that data-driven open science offers an opportunity to develop innovative biofilm-based technologies for both deep space exploration, but also meaningful impact on Earth. Next slide, please. And that's the kind of overview of our perspective manuscript. I'd like to thank everybody for listening, all the biofilm subgroup members that contributed and made this manuscript possible, all the microbes analysis working group members that kind of supported us, and also the Genelab, OSDR and LSDA team members who, without being here, we wouldn't be here. And I'd also like to plug our next meeting, which is next Tuesday at 10 p.m. Indian Standard Time, 5:30 p.m. British Summer Time, and 9:30 a.m. Pacific Time. Thank you.
Hello.
Yes.
Okay. Well, hello everyone. I'm Aurora Diaz. I'm a physician and bioinformatic researcher. Today, I will introduce our project on the molecular landscape of viral reactivation in Alzheimer's disease and space-like induced stress as part of the AWG Brain Group. This work is part of a collaboration with Bruno Pavletich, Dr. Nidia Trovaud, and Professor Dr. Margaret Wendy McNerney. Next slide, please. Well, as we know, Alzheimer's disease is characterized by protein deposits like amyloid, beta and tau proteins, and chronic microglia driving neuroinflammation. These factors can weaken the blood-brain barrier, making the brain more permeable. When this is combined with chronic systemic immune suppression, conditions arise that can allow latent viruses, such as herpes virus 1, to reactivate. Once active, these viruses can contribute to the production of amyloid beta and tau proteins that could drive to Alzheimer's pathology. And as we see also in spaceflight, systemic immune suppression can be caused by environmental stressors such as microgravity, radiation, isolation, and these stressors can reduce the immune surveillance. And this can also promote neuroinflammation. Now, when chronic neuroinflammation and chronic immune suppression coincide, the brain becomes more vulnerable and viral activity may accelerate pathological processes. So even though Alzheimer's disease and spaceflight are very different, both involve stressors that converge to promote viral reactivation. Next slide, please. Okay, well, now let's look more closely at the immune dysregulation. Under stress conditions, such as spaceflight, the immune system shows both pro-inflammatory spikes like TNF-alpha and interleukin-6, and reduced anti-inflammatory responses, for example, interleukin-10. Also, the T cells and natural killer cells become less effective, weakening the immune surveillance. In the brain, this imbalance is mirrored by microglia, which are normally in a resting state, but can polarize towards an M1 pro-inflammatory or an M2 anti-inflammatory phenotype. This switch is observed both in aging and neurodegenerative diseases on Earth and in astronauts exposed to microgravity, radiation, and stress in space. And also, as we know, microglia is like the resident immune cell in the brain, for example. Well, such dysregulation ultimately impacts neural health and creates conditions that allow latent herpes viruses. For example, herpes virus 1, Epstein-Barr virus, and varicella zoster virus, and this can lead to escape latency and reactivate of these viruses. Next slide, please. Now, for example, when herpes virus 1 reactivates in the brain, it directly triggers neuroinflammation. In brain tissue models, this reactivation has been shown to induce protein clamps characteristic of Alzheimer's and activate pro-inflammatory immune cells. As we see here, it also drives oxidative stress and NF-kappa beta signaling, leading to hyperphosphorylation of tau proteins. At the same time, herpes virus 1 increased beta and gamma secretase activity, which promotes amyloid beta peptide accumulation. Taken together, the herpes virus's reactivation contributes to both major hallmarks of Alzheimer's disease, as for example, the presence of amyloid beta plaques and tau tangles. A similar risk environment may also occur in spaceflight due to immune suppression and neuroinflammation. Next slide, please. This project is made of two parts. In both, we analyzed RNA sequencing data from peripheral blood. And first, we identify viral reads among the sequences and quantify them to determine virus reactivation. And second, we analyze human gene expression to model and quantify pathway activities. Together, the viral reactivation will be correlated to the pathway activities to determine the most significant pathways connected to virus reactivation in Alzheimer's disease patients and astronauts. For the astronaut data, the RNA sequence mainly came from and this contained inspiration for mission astronaut blood RNA sequence data. And later we will move to other OSDR datasets. Now Bruno will continue with the presentation. Thank you.
Thanks, Aurora, for this wonderful introduction. So now with the virus detection part, so we started with RNA-seq reads, as Aurora explained, from Alzheimer's disease patients and cognitively normal controls. This is based on the data from the study of Stu et al., whose reference you could see on the previous slide, and the data was comprised from 3 Alzheimer's disease patients and 2 normal controls, and each in the end each of the samples had 4 technical replicates. We identified the viral reads by searching against the virus coding sequence database. Then we assembled viral genes from the identified reads. And finally, in the end, we will quantify the reads, quantify the viral reads that were identified per each sample. Next slide, please. Then it was one, then for the gene expression part of the project, we mapped the gene expression values from patients and controls onto their KEGG pathways. Biological pathways can be described as circuits of information, and therefore the information flow in the pathway can be quantified based on the abundance of each node in the network, and each node is represented by one gene. right? Protein abundance can be assumed from gene expression. So ultimately, pathway activities of each pathway can be quantified. Finally, the gene and pathway activities will be correlated to the viral reactivation in Alzheimer's disease patients and astronauts. Next slide, please. Yeah, so to come to the results part, from the virus detection, the part of the project, we identified many different viral genes in Alzheimer's disease patients, which still remain to be quantified. There were also some in the control, but here is one example of one viral, even viral RNA genome from human endogenous retrovirus K113, and which was assembled from this data. Once we identify all the different viruses and all the different viral reads in each sample, we will proceed with phylogenetic analysis, which will be aided by media. And here you can also see some of the example viruses that were found in among the samples. Next slide, please. Yeah. So from the gene expression part of the results, we determined the gene activities and plotted them all together. On the left, so on the left side, you can see the normalized gene counts from each sample. Of course, every sample had the 4 technical replicates. That's where you see 20 samples, and then you can see in the middle the heat map of pathway activities, clustered by similarity and by sample. The red pathways, so you can see the pathways on the left side of the heat map. So the red ones have higher activity in Alzheimer's disease compared to the control, while the green ones have lower activity in Alzheimer's disease. And finally, on the right side, there is a PCA plot based on the pathway activities, and the samples, as you can see, cluster very nicely together. And those samples show very good, very high difference considering they come from blood, which is very nice because blood usually doesn't show so much difference. So there could be potential of using pathway activities as biomarkers for Alzheimer's. Next slide, please. And finally, just to show you how this mapping looks schematically, here are 2 example pathways that showed higher activities in Alzheimer's disease than the control. One of the interesting pathways connected to viral reactivation is herpes simplex virus reactivation pathway. And also considering inflammatory stress and immune activity, NF-kappa B signaling pathway is also interesting to see here. And you can see that some genes are upregulated and the others are downregulated. And when taking into account overall expression, the pathway activity can be determined from each and compared between the samples. So please, next slide. So in conclusion, we developed the pipeline based on the data from Alzheimer's disease patients. However, first, the completion of the analysis of Alzheimer's disease is in place. Then, second, we will get the RNA-seq data from the astronaut's peripheral blood to also be evaluated through the same pipeline. This is in collaboration with Dr. Jung-Keun Kim and Professor Christopher Mason, and the correlation will be established and the differences and similarities between the astronauts and Alzheimer's disease viral reactivation taken together with the gene expression. So correlation between gene expression and viral reactivation. And right now we are preparing the manuscript considering the pipeline. Next slide, please. Thank you very much for your attention. Thank you.
Hi, thank you. Thank you for the introduction, Ryan. As Ryan said, my name is Matthias Nesset. I am a PhD student at McGill University. And thank you for all coming today to listen to this talk. Today I'll be talking about a project that's, like Ryan mentioned, has been a long time coming in this ALSDA-AWG, and it's titled The Heart Rate Outcomes in Humans Exposed to Microgravity, a Systematic Review and Meta-Analysis. Next slide, please. All right, so to start, I'll just give a little bit of background. So as I'm sure most of the people here know, spaceflight is a very hostile environment, and it's also characterized by several significant physiological changes, including muscle loss, bone degradation, mental stressors, the list goes on. And so NASA already identifies 5 key hazards for human spaceflight, including radiation, microgravity, which is the focus of today's work from the title, isolation confinement, closed hostile environments, and distance from Earth. So with looking at all these hazards and barriers, this means that spaceflight experiments are actually relatively rare compared to most general other experiments. And this makes the data that they publish ever so important for, drawing those further conclusions. However, oftentimes these data that they do release often come with very small sample sizes, making large generalizable conclusions about the outcomes more difficult. So what you can do to kind of counteract this is you can start to look at something like translational models. This could be like on Earth models, other populations, so on, or even things such as Earth-based diseases that have similar effects, like in muscular dystrophy and osteoporosis, where there's similar microgravity unloading effects. However, these also come with their own limitations. But there is also one big thing that is commonly known, and that is knowledge synthesis. And that's where you take all the published literature and put it together in order to produce something that with stronger power and in order to allow you to better interpret the results. Next slide, please. So looking at some of the cardiovascular changes that occur, it's generally agreed that the cardiovascular changes include, changes like alterations in cardiac output, decreases in blood pressure, cephalid fluid shifts, which is the fluid shifting towards the head and upper body. And this can lead to other complications. One of the more common ones that's very focused on now is known as SANS, or spaceflight associated neuroocular syndrome, where you get increased pressure in the eyes. However, despite this general agreement on all these changes that occur in the cardiovascular system, there's still much to be learned about the complex interactions either between these specific cardiovascular outcomes themselves or their implications on other physiological systems. So upon further investigation into this topic, in the literature, we found that there's actually no large grouping of cardiovascular data, but there are thousands of potential articles available. Next slide, please. So that leads us to our objective for this project, which was to quantitatively analyze heart rate related outcomes amongst cardiovascular outcomes in humans exposed to actual microgravity. And this is done through meta-analysis. So here we have a bit of a prelim to this, which is the systematic review part. Today I'll just kind of skim through it as this is kind of its own thing on itself, and I can give a whole talk on this. But so just to summarize, what we did is we performed a systematic review to identify key cardiovascular articles. This was done with the help of AWG members to help identify specific key terms and go through some of the things that we should be looking for. And then what we also did is we also developed the Solstice, or the Space Open Life Science Team for International Collaborative Exploration, which is a citizen science program that allowed anyone from any background to contribute to the research we were doing, which gave them access to doing spaceflight research, and which also helped us move along our timeline much quicker. As you'll see in the next slides, that there's quite a few articles that we did. So overall, the systematic review, I won't get into nitty gritty, like I said, but we ended up summarizing the existing cardiovascular articles that exist in microgravity. And what we did is we then divided them into several subgroups for further analysis. Next slide, please. So here we have the Prisma diagram, which effectively outlines the process in which we did our systematic review and how we got to the articles that we used for our meta-analysis. There's a lot of information here, but what I kind of want to draw your attention to is those 3 main arrows. So the one on the top, you can see we started with just under 19,000 articles, which for anyone who's done any sort of publication review or like literary searches, that is an extremely high number. And so that's why we actually ended up having the benefit of using that citizen science team to help us do this project in a relatively manageable time frame. And so what we did is we then got those articles down, we title and abstract screened them, and then we subdivided them into groups based on different outcomes. Today, we'll be focusing on the humans and actual microgravity group, where after full text screening, we ended up down by the third arrow with 189 articles for inclusion. We then went a little bit further and subdivided them into specific cardiovascular outcome groups, where we'll be talking about the 70 heart rate articles there. So next slide, please. So just to give you a little bit of preface into how we did our analysis, what I want to outline here for the heart rate values, as you can see on screen, these are just straight up averaged values, just so you can see some comparisons between groups. But things I want to outline are the 3 measurement time points. So we had generally pre-flight, in-flight, and post-flight groups. Measurement positions were very dependent on, heart rate values were very dependent on measurement positions. As if you look just at the pre-flight values, you can see the standing and supine are all drastically different. So we subdivided them into subgroups based on measurement position as well. However, for the in-flight values, we just aggregated these data from those measurement positions together due to the lack of gravitational vector. So all values were assumed to be taken in a similar position. Next slide, please. Just looking at the analysis that we did to compare the groups. So what we did is we included any studies that reported at least 2 groups. We used a standardized mean difference model, and we did our analysis using Bayesian analysis, using a fixed effects model. So the reason we chose this is because it provides a better accounting for smaller n values, which is kind of the popular issue with spaceflight data, and it helps describe studies that have similar environment and similar stressors. So the results that I'll be showing today will be expressed via a forest plot. I'll describe it a little more when we get there, and the equations were there. So we're looking at the first set of values. So just looking at the heart rate values themselves, we have the pre to in-flight changes. So these are the changes between the values pre-flight to in-flight. Overall, across all of the other groups, so here we have supine, other includes both seated and standing, and then we have the unspecified groups. There's no change overall, and this is characterized by the dotted line in the middle with the 0. Those big diamonds represent the summary of each group. So the big diamonds on the screen, as long as they cross that 0 line, it indicates no significant change in any I just want to outline in the pre to in-flight. So in-flight, there was no significant change in heart rate values. Next slide, please. However, once we move to pre to post flight, as you can see, not only does the actual number of articles reporting this increase, so you can see there's quite a bit less in-flight data compared to pre to post. But what you can see here is, if you actually go to the next slide, Ryan, I will have highlighted just to draw your attention to those summary diamonds of the specific groups. And as you can see in the supine, standing, unspecified groups, so 3 out of the 4 groups, there is a significant increase in heart rate values from pre to post flight with quite large effect sizes. You can see you have 0.99, 1.92. So these are deemed very high effect sizes. So there's a very significant increase in these values. And even if you look at the global effect, which is the last red box on the screen, this accounts for all of the measurement groups together. You can see there's still a significant increase overall in that. So here we have just from pre to post flight. So now what we wanted to do is we wanted to take this analysis a little bit further, and we wanted to look at specific if there were any covariate differences between the outcomes. So what we did is on the top row of A, B, and C, these were the pre to in-flight values, and on the bottom row D, E, and F, you have the pre to post-flight values. The first column, so A and D, what we did is we assessed the heart rate outcomes both are depending on mission platform. So we separated and we recorded which mission platform these studies were reported on. And as you can see here, we can see the differences. So most notably in A, so the pre to in-flight values, you can see that in the ISS, there was a significantly higher, or rather in the shuttle missions, there's a significant decrease in heart rate values. And this is actually mimicked over into the B plot, which looks at the average time in space. So here the data is subgrouped into 2 groups of a short-term flight, which is under 50 days, or a longer-term flight, which is over 125 days. And here you can see a very similar pattern between the shuttle and ISS missions, where the shorter-term flights have a significantly decreased heart rate compared to those of the higher, longer-term flights. Now, one caveat with this is if you go back, yeah, no worries, is that we did notice that shuttle missions do tend to be shorter. So there can be some kind of combination in that. However, we believe that this should be investigated further. For the pre to post flight values, there were no changes in both mission platforms or days in space. And then if we look at the actual C, so what we wanted to do is for figure C, we wanted to investigate these specific differences a little bit further. And what we did is we analyzed and we followed the groups, or each individual group's variation during their in-flight values. So the plot here looks at the variation in heart rate during their time in flight. And the key thing that you want to take away from here is that the shuttle groups, while they're all on the left in the early flight days, they all have that significant decrease start and then it slowly average normalizes out to similar to what the ISS values have. So long term, it seems to be very little effect, but in those very early days is where you kind of see that decrease, and that's something we noticed, but there's very limited data in that time frame, so we think that's something that should be investigated further. Just one final note on plot F, we investigated some of the recovery times. So as you can see here, upon returning, the values are typically quite a bit higher than what they were pre-flight, but after approximately 30 days, these values in all the groups tend to return back to normal. Next slide, please. So now moving on just from the specific heart rates and the typical heart rate beats per minute that you were thinking of. Now we went a little bit further and tried to describe some of the heart rate variability. So specifically, heart rate variability describes time between consecutive heartbeats. This is done through various devices, and here you have just a variety of things that they can mean, such as HF, high frequency, meaning parasympathetic system, and then beat to beat is RR intervals. Next slide, please. So this is just all of the heart rate variability parameters that can be reported. And so what we did is we looked at these and we counted the number of times these were reported in the articles that we had included. And if you go to the next slide, please, Ryan, you can see here that we ended up including the values that were most commonly reported. And this aligns with literature that we compared it to, which recommended reporting in these parameters. So next slide, please. So if we look at, again, the pre to in-flight values, I won't spend too much time here. In all cases, in both the time domain and frequency domain for parameters, there was no significant change across any groups. If you go to the next slide, please. However, again, in a similar fashion, we had if we look at the pre to post values, as you can see by the title on the slide, in the top left, we have RR, in the middle on the right, we have HF, and in the bottom right, LFHF. All three of these groups showed significant difference. I believe if you go to the next slide, I should have just the right, yeah, here. And so what this shows is, so here the RR values significantly decrease. Since RR is actually directly proportional to HR, this aligns with the other data that we had with the heart rate being significantly increased. Therefore, the time between the beats, which is the RR interval, is significantly decreased. So this kind of confirms the analysis that we found in the first part. But if you look more into the right side at those frequency domain values, you can see the HF, which is the high frequency, which represents more of the parasympathetic side. It is significantly decreased, and as a result, the LF over HF, which is a ratio of the top two, is increased, which what the data showed, and it follows mathematical proportions. Next slide, please. We wanted to do further cofactor analysis in these groups. However, we looked at all the significant groups across similar effects. So in the top here, we have the 3 mission platforms, and the bottom, we have the days and space. However, across all the groups, there was no significant change for this group or for these results. Next slide, please. So just looking at some of the physiological relevance of this information. So for the increased heart rate side, the increased heart rate implies a higher workload. There's been links, shown before with orthostatic intolerance, and this is quite commonly studied, where heart rate has these links, and this is kind of where I outlined at the start the need to further understand the complex interactions between specific outcomes and specific measurements to understand further how specific areas might change. And this is so that then it's also increased with cardiac output demands. So more when you come back to Earth, you're resubmitting the heart to more gravitational loading, so a higher cardiac output, and then implications with lower stroke volume. So does this correspond to a higher heart rate, and so on. Additionally, there's been studies that report increased musculoskeletal demand, which can increase the heart rate. And like I said, the decreased RR values kind of support our heart rate increases. However, when you look at the heart rate variability side, some of the physiological relevance to what it actually means is that the decreased high frequency values indicate a reduced parasympathetic activity, which kind of implies that during that time frame, astronauts will likely be more stressed, worried, have more difficulty recovering in that more relaxed state, and this can lead to increased fatigue. And it actually has been known on Earth to have implications in other cardiovascular diseases, such as being a predictor for hypertension, heart failure, and stroke. So this is something that should be further analyzed to do that. So while we were able to determine these kind of physiological relevance and everything, we are open to hearing more about just expert interpretation on these results, just to see what else other people might think. And I put AWG there to hear from what you guys might think about what this might mean, implications, and so on. So next slide, please. So just to go over the limitations of the study, some of the limitations we ran into is the aggregation of data. Most studies did not report individual data points, probably 95% due to just ethical considerations. So we can't do any further age or sex analysis. We can't do any subgrouping based off experience fitness level. And we cannot necessarily guarantee data reuse between studies. However, we did try to account for this in the best way that we could. Additionally, we had mission characteristics weren't always available, so we had to limit them to mission to platform data. And we had the limited in-flight. So overall, just to go over the key things that we saw, we had no in-flight changes across any groups. We had a potential significant decrease in heart rate during the early flight days. However, more data is needed to analyze those specific time points. Most groups experienced significant change between pre to post flight. All of the heart rate groups significantly increased. We had the heart rate variability decrease in RR values and the high frequency, so the parasympathetic response, and then the resultant increase in the LFHF ratio. And then finally, heart rate returned to pre-flight values after 30 days, meaning it is somewhat recoverable after the spaceflight, unlike other systems, which like bone and muscle, which can take much more longer periods of time. And then I believe next slide will just be the acknowledgements for everyone who's helped work on this project, from my PIs to my collaborators, including Ryan, the AWG members, Solstice members, and my lab. Thank you. Thank you.
Thank you, Ryan. Hello, everyone. My name is Jessica Durbano. I'm an Italian medical doctor, currently working in the field of occupational and space medicine. And today I'm very happy to share with you our AOP, which is called the position of energy leading to decreased fertility. I am the co-lead along with Mimi, and our point of contact at Health Canada is Venita Chawan. Next slide, please. So what's an AOP? An AOP is a hypothetical construct that connects essential key events, also called keys, from a molecular initiating event to an adverse outcome. The AOPs are living documents that reflect the current state of knowledge and are ever evolving. AOPs are not stressor specific. They do not have regard for which specific stressor initiated a response. So an AOP consists of a main initiating event, which is the first chemical or physical interaction with the body, the key events that are measurable, most biologically dominant, is essential to achieve the adverse outcome. The key event relationships, causally linked and supported by empirical evidence, and finally the adverse outcome, which is the phenotypic change. So we are starting from an insult, going on to molecular interaction, which can be receptor-ligand interaction, DNA binding, or for example, protein oxidation. Then there's a cellular response, gene activation, protein production, alter signaling. Moving on to organ responses, alter physiology, disrupted homeostasis, alter tissue development and or function. Organism responses, lethality, impaired development, and impaired reproduction. And last but not least, population responses, which could be, for example, extinction. Hopefully not. Next slide, please. So how do we build an AOP? To build an AOP, one typically does literature collation and filtration. So literature collation and filtration helps identify the key events that connect to other key events and then leads to the proposed adverse outcome, the AO. The linking of the key events is a key event relationship. These key event relationships are directional and causal, so they are supported by a weight of evidence. The weight of evidence consists of primary research data that justifies the strength of evidence of the key events relationships. So we collate literature, we filter studies, we identify the key events, and we identify the linkages between the events. Next slide, please. For building an AOP, it's essential to use the modified Bradford-Hill criteria. I think that a lot of you know the standard Bradford-Hill criteria, but in this case, we use the modified ones. They evaluate the strength of causal linkages between the key events relationships. And these modified criteria are 4. We're moving from the strongest to the weakest. The strongest is biological plausibility. So the key events relationships are consistent with current biological understanding. Then there's the essentiality of key events. Are the effects reversible if the stressor is removed? Yes or no? Then there's the concordance of empirical observations. So dose response. Are the key events observed at doses below or similar to those associated with the apical effect? Temporality. Are the key events observed in hypothesized order? Incidence. Is the frequency of occurrence of the apical effect less than that for the key events? And last, which is the weakest, consistency. Is there agreement in data across experiments? So we're basing on the we based our literature on the modified Bradford-Hill criteria. Next slide, please. What is our AOP about? We decided to focus on female infertility. We know that infertility is a disorder of reproductive system which is characterized by the inability to achieve a pregnancy after 12 months or more of regular unprotected sexual intercourse. The causes of infertility can be linked to congenital or acquired pathologies of the reproductive organs and or to disorders of the endocrine system. It is already known in literature that ionizing radiation has a gonadotoxic effect, acting directly on the uterus and ovaries or disrupting the hypothalamic pituitary gonadal axis, so disrupting the endocrine system. The long-term effects could include ovarian insufficiency, pubertal arrest, and subsequent infertility. How do we relate reproduction with space? Understanding the effects of and prolonged space radiation, both in short and long periods of exposure, on the functionality of female reproductive organs is crucial. This is what we are trying to focus on. This also includes evaluating the overall impact on cancer risk, not only on the progenitors, but also on the future generations through effects on their gametes. Moreover, it is important to differentiate the impact of radiation versus the aging influence, which naturally affects reproduction. Next slide, please. Which one was the state of art in 2024 last year when we started our AOP? So the available literature in these areas is not very extensive. Most of the studies are conducted using laboratory animal models. In addition, evidence is extracted from past missions in low orbit, especially experienced in the ISS and the International Space Station. Next slide, please. This is our hypothetical AOP. So as we've seen before, we're starting from a macromolecular point of view, moving on to cell, tissue, and then individual. Our main initiating event is deposition of energy. Then we're moving to key event number 1, increase in oxidative stress, and moving on at the same time to key event number 2, increase in DNA strand breaks. Both are related to the key event number 3 at cellular level, increase in oocytes apoptosis. Then we're moving on with the key event relationship to a tissue level, key event number 4, reduced follicular pool. At individual level, we reach the adverse outcome, which is decreased fertility in females. Next slide, please. But why create an AOP for infertility? An AOP it's a standardized framework to organize mechanistic evidence. It bridges animal and in vitro data to human health outcomes. It helps us identify critical knowledge gaps to guide future research and supports regulatory frameworks and risk assessment needs, for example, radiation protection and space health. So these are the main reasons for why we decided to build this AOP. Which are methods and deadlines. We are currently using the Stiller, which is a screening software, and library resources for the development of the AOP using a scoping review methodology. We started in December 2024, and our goal is to submit our AOP to the OECD Secretariat for endorsement by December 2027. Thanks to the Genelab Multiomics AWG that provided us students and also the SMEs that are the subject matter experts that are crucial in the development of our AOP. We are currently at level 3 data extraction. Next slide, please. So Mimi and I are the co-leads. As I said before, I'm a medical doctor in occupational and space medicine field. Mimi is a master in health science at Johns Hopkins University, and Benita Chawan is our point of contact. I'm very thankful to her. I'm also very thankful to Mimi. We have as subject matter expert, Dr. Edward Azam, Dr. Jean Kaleha-Ajus, Dr. Ruth Farrell, Dr. Begum Matic, which is our female AVG lead, Dr. Haley Reyes, and Dr. Marcelo Vasquez. So thanks to all of them, and also thanks to Sadia, Paul, and Bianca that are really, really crucial in all the phases, especially now during the data extraction. Next slide, please. To conclude my presentation, I would like to read a few lines that my co-lead, Mimi, wrote as a student perspective. So she said, "My experience on the female reproductive AOP project has been so incredibly rewarding. This project has refined my research skills and critical thinking. I am learning firsthand how to develop and successfully execute a research project from start to finish. Additionally, collaborating with such passionate people such as Dr. Vinita and Dr. Jessica has greatly enhanced this experience. I helped modify the study protocol, manage the student working group, and complete data extractions. Currently, I am helping to refine our search strategy for the literature on our various key event relationships within our AOP. My main takeaway thus far is that everything is a work in progress, and we must be flexible and dynamic in our approach. There may be obstacles, but in those challenges comes the most growth. I wanted to thank Dr. Locke and the Johns Hopkins Toxicology team for their support." So these are her thoughts about our AOP. So thanks Mimi, thanks Vanita, and thanks to all the Multiomics AVG and to all the other AVGs for the collaboration. Next slide, I think, is the last one. Thank you. Thank you all.
Yeah. Can you all hear me? Awesome. Yeah. Thanks, Ryan. Today, hi, I'm Vaishnavi, an ML engineer and a bioinformatics scientist at Genentech, and today I present adaptive imputation method that we developed as part of developing a digital twin for mice retina. Our research demonstrates the utility of these methods in building a digital twin for SANS, or space flight as associated neuroocular syndrome. It's one of, like we saw earlier, it's one of the high priority health concern for astronauts. A big thanks to all the digital twin members subgroup, especially Gian, Lauren, Ryan, Sylvan, and I wanted to make sure I get the acknowledgements first in case I run out of time. Moving on to next slide. So a little bit about what the digital twin project goals are and how we arrived at the imputation subgroup goals, sub-project goals. Sorry, give me a second. So basically, the reason to create a digital twin in the mice retina space was to kind of identify a bunch of what if analysis by manipulating features and examining phenotypes. And one of the challenges in creating a digital twin was actually having a very sparse data set. And that is where the imputation sub-project goals came into picture. And this is something very common that we have seen in space biology, the challenge of incomplete data due to logistical complexities, high costs of space missions, and that is where this adaptive imputation method comes into picture. So by presenting a cohesive strategy that combines synthetic data generation, automatic labeling, and advanced machine learning with digital twins, we showcase an application to the RR9 dataset at the OSDR. So a little, if we go on to next slide, if I think there's one slide in between. Yeah. A little bit background into the kind of data we worked on or we are continuing to work on, and how we plan on making this go ahead with imputation and digital twin technology itself. So, in general, spaceflight drives unique vision and joint risks, and it demands multimodal analysis. And one thing about RR9 dataset is that it investigated that RR9 mission investigated how long duration spaceflights affects vision and joint health in mice, modeling astronaut risks. So there were three main experimental themes. One was fluid shifts and intracranial pressure, which examined vascular and lymphatic changes in the brain and eye caused by microgravity. Then retinal, vascular, and oxidative stresses. And the third one was joint degradation. Our work is focused on ocular and retinal investigations, and the data sets being explored are OSD 255, 568, 557, 583, and 715. Something to note here is in all of the work that we present, this one and future works too, it's actually combining multiple assays, which includes gene expressions, proteomics, immunostaining, and tonometry data, micro-CT, and whatnot. It contains substantial missingness across several assays, except tonometry, which we have a complete data set, and it actually qualifies as a case study for sophisticated imputation and digital twin development. Next slide. So, jumping in, the first heat map kind of tells you how many data sets we have and against how many categories of data that we have, which data sets are missing. So overall, we have 100 mice, 20 in flight and remaining in ground control and vivarium divided as two cohorts. So the first heat map shows you that we actually have a complete tonometry data set. And you can see that for some mice, for some categories of mice, especially like cohort 1 and 2 and vivarium, RNA-seq is missing, H&E data sets is missing, and we wanted to get an idea of how much of data is missing. And this kind of tells us we have around 85 to 90% data missing. The second is something called a nullity correlation matrix. It's generated by calculating pairwise correlations of missingness between variables. So instead of correlations of their observed values. So what this shows is whether the missing values in one variable tend to coincide with the missing values in another. And if if you notice, there is a strong missingness cluster among assays like PCHEM, PNA, tunnel, HNE, and RNA-seq, often missing together due to probably due to shared experimental steps or sample loss. In contrast, if you see microCT and protein actually show independence or weak correlations to their missing patterns. Something to note here that probably will not, unless we kind of make it explicit, you might not notice is this is actually just 100 samples, but it's a very, very high dimensional data set. It's around 7,000 features that we are working with. So this is actually probably an eagle flying at 13,000 feet view of everything that we have worked on. Next slide. So before we get into imputation, we want to identify how is the missingness being treated? Like where is the missingness coming from? Is it because the data is just missing at random, which can happen? Sometimes samples get lost, or sometimes we just don't have enough samples, or is it missing completely at random, which means that probably some assays were deliberately skipped because we didn't want we didn't want to record some assays, or it was missing not at random, wherein there was just I don't know, there was just no goal to set up assays for all the samples and everything. So there is a small statistical test called Little's MCAR test that either accepts or rejects the hypothesis that data is missing completely at random. And MCAR is slightly important here because we should not be imputing any data where the data has been deliberately missed. Or rather, if the data is missing completely at random, then we basically don't impute them at all. So we used this Little's MCAR test and logistic regression to check if missingness was independent and if there were any violations for the MCAR assumption. And what it told us was for most of the data that we are seeing, everything is basically missing at random except tonometry. We saw earlier there is actually no missing data. The logistic regression and the deviations or any deviations from MCAR hypothesis also kind of gave us a few practical implications. It set us on a path to use different multivariate methods to identify whether we can simply use mean, median, or mode to impute values, or if we need a more comprehensive and adaptable approach. So a key takeaway from this slide is values are missing at random, so we went ahead and used multivariate methods, and that's what would be our next slide. So if we remember from the nullity matrix identified like probably 2 slides ago, we identified that some categories were preserving very strong clusters of missingness. And we went ahead and created an endogram out of it. And to show that clustering kind of reveals which assays could be imputed together and which assays should not be imputed together. And this kind of helped, instead of simply throwing in a bunch of data sets against a different imputation algorithm, you're going at it in a very statistical way. Things that if PCHEM and ZO1 are constantly, co-occurring or co-missing together, then you know that those values are highly correlated. And you can use this dendrogram to kind of go at it in a stepwise manner. So the immunostaining assays, PCHEM, PNA, ZO1, tunnel, HNE, and RNA-seq in that order consistently cluster together. But if you notice protein, microCT, and tonometry, they consistently cluster very separately, which means at least proteomics and microCT data need a very different kind of imputation methodology. So we took this we took this whole dendrogram a little further, and what we threw the entire data set, the 7,000 features data set, against clustering and determining the nullity matrix, and saw that some of these, for example, these different microCT, they co-miss together, which is obviously like if you don't have a right microCT value, you obviously don't have a left microCT value because probably the sample didn't have microCT assay done at all together. So this was kind of what our anchoring was for our imputation methodology. And it told us how we should go about imputing each feature with respect to an earlier present feature. The second, the horizontal dendrogram kind of, is our starting point to anchor our imputation approaches. Next slide. So there were multiple methods that we did. One of the key methods over here is KNN, which is like the simplest and very quick to run. Second method is a slightly complex approach. It's called iterative imputation or MICE, multi imputation with chained equation. It handles more complex patterns by modeling each feature in turn. Think of it like running a linear regression with every feature one at a time. And then mice can be implemented for different kind of regression techniques. The first one was Bayesian ridge or bagging regressor. We used both of them. There was also a boosting regressor with iterative imputation, and one of lesser acknowledged approach is random sampling imputation, which is generally used for very small data set, but we didn't want to let it be alone. And then we tried something called a hybrid approach, which is what a big takeaway of this whole talk is that it's adaptive. So one of the reasons we took a hybrid approach was as we got to the validation aspects of imputation and everything, we saw that some of these approaches, for example, KNN or RSI, the random sampling computer would run really fast, but it would not preserve the coherence in the data as the original data. But some of the slower methods like iterative imputation, they would probably take days to get to results, but it preserves the correlation in the data set better. So, and not to forget, we have 7,000 features to impute. So we were like, okay, how can we get this faster? And that's where the clustering, dendrogram, and this hybrid approach kind of came together. One of the big takeaways of exploring different methods is we need to escalate the imputation model complexity only as needed. Definitely not everything has to go through the highest algorithm out there. Validation and masking unreliable imputations at each step is important. That was not something that we did at the KNN step. But as we started developing our hybrid and adaptable approach, it's something we saw it was very valid. So we could take out any data that wasn't meeting, our expectations of RMSE thresholds or MAE thresholds, and impute it with a slightly complex approach. Next slide. So our hybrid imputations approach, the heat map on the side kind of shows you that we started out with, KNN, and we kept a low threshold of less than 2. Actually, the heat map is slightly out of date. But with KNN alone, we were able to impute around 6,684 features, and whatever didn't meet our RMSE thresholds were run through the mice, slightly complex iterative imputer approach with Bayesian ridge regression. There we imputed 4 more features. The remaining, features were taken through the boost regression approach for imputation. And then the remaining features again were taken through the bagging regression. And finally, at the end, we had like 6,692 features imputed. And this was something that was really fast compared to just a brute force iterative imputer alone, and more accurate than just random simple imputer or KNN imputer alone. One of the biggest advantages is that it adapts to the model complexity, to data challenge, and it ensures speed and accuracy. And most importantly, it actually preserved the critical biological and feature relationships that we had originally seen using PCA and different correlation metrics, and it outperformed every single method approaches that we had tested against. Next slide. So we have finished imputation. So what's next? We are talking about validation as we go during the imputation, but there still has to be something that ties everything together before we go into development of digital twin, because we want the digital twins to be very much representative of flight and non-flight, and as we happen to see in some of the PCA analysis, that was too short of a time to present here, but the signals were not extremely clear. There was a clear separation, but it was not as clear as sun and the moon, probably. So we did develop some rigorous validation approaches. And one of them involved doing a supervised classification like SVM and random forest, and then an unsupervised k-means clustering to see how well the groups actually cluster away from each other. There was also identifying whether the correlation structure, basically the pairwise feature relationships between different features in original data, do we recover the same pairwise correlations in the imputed data? And then a statistical test to say how well does the original and the imputed values to the distributions, how well do they match. Present the results in the next slide. So one of the things, actually, it goes a little in a reverse chronological order, but one of the last things we did was also visually seeing if they separate well. So the first one here is actually a PC of the original data. And you can see that if there is some difference, like you can draw probably a line between the ground and space data sets. When we did the multiple imputation with chained equation using the bagging regressor approach, we saw a lot of good things come out through SVM, but when we actually did a visual inspection, they weren't, you couldn't tell the difference if I had not colored the dots. But with our adaptive approach, there is still some artifact left, not to deny that, but you do see that the space generally separates away from the ground. There's definitely more tweaking to the algorithm that we can do, but it's at least closer than what we were seeing in KNN and other approaches. So overall, for the validation data set, in general, what we can see is with random forest, our hybrid or adaptive approach actually has 93% accuracy in separating space and flight and non-flight samples. Clustering does reduce, our clustering ARI scores do reduce, and there are ways to accommodate that. The correlation metric, which was something that I was looking for, and the mean KS stat is closest with the original data with the hybrid approach as compared to the other two imputation approaches. Next slide. So what recommendations do we go home with after all this? So in general, using a hybrid multivariate imputation, like cascading it down from KNN and then Bayesian Ridge algorithm and then XGBoost algorithm, and accepting only high quality columns at each stage of imputation actually is a better approach than one single method of imputation. There has to be there has to be a very rigorous validation of imputation approaches, which has to be multifaceted, mainly because we have a very high dimensional data set. One of the things that we should always look for is we preserve transparent missingness, where the columns cannot be imputed reliably. We need to leave values as is, as opposed to forcing imputation and kind of misrepresenting our data. With respect to data sets where we have such a non-random missingness and systematic missingness, in general, we need to avoid simple imputation approaches. Definitely, we need to during the development of this whole imputation method, one thing we did observe that we need to keep benchmarking the imputation performance with strict RMSE and MAE thresholds, and only accept features that are passing the quality control for downstream digital twin modeling. Next steps, we do have a manuscript in progress, and the next step is actually to develop a digital twin.
Hello? That was it. All right, I think we're back. Can someone give me a mic check on YouTube? And I'm assuming the room can hear me. Maybe I can just get a thumbs up from someone in the chat or something. But I think we're good because next up is going to be Borja. Yeah, Jennifer, if you could double check on YouTube that this is indeed the audio, I believe, is now rectified, I think. Let's see, let me find Borja. All right, so we got Borja. Excellent. Borja, can you hear me? Borja, can you hear me? Uh-oh. Did I mess something up? Borja, can you hear me? Oh. Well, that's not good. Shoot, what did I do? It says I'm muted, huh? Shoot, let me find out what's going on. Unmute. What happened? What did I do? Can anyone hear me now? I think I might have messed something up. Let me see if I can... I don't know if that's going to take care of it. Oh, you can hear me through YouTube now, but not in the meeting. Oh, what is going on? Here, let me try one more thing then. I'm super sorry about this. Is this audio working? Borja? Oh man, what is going on? Oh no. Shoot. Hello? 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 Now I can't hear. Oh man, what's going on? Trying to get happened? I'm gonna try one more thing. Can you hear me? Borja? Can anyone hear Borja? I guess that's important. Actually, yeah, maybe Amanda, could you go to the podium? Can anyone hear me? What happened? Oh, shoot. Yes, Amanda, go for it. Oh, shoot. That's the display. Oh, that's super weird now, Danielle. Mic auxiliary. What happened? The settings were all set. I can't even hear now, though. It's like everything is muted. Oh my gosh, I figured it out. Oh, I'm so sorry. I figured it out.
Technology.
Yeah, there was a mute button on your profile that didn't automatically go away. Gosh darn it. Well, thank you for. I'm sorry about the false start of all the times. I was all dialed in. Okay, I think everyone can hear me. Maybe can hear you, Borja, and that was just because of this mute button on your profile. Okay, whatever. That was my bad. Welcome, Borja. I'm very excited about this talk and what the plant, AWG, BioAstra, ALSDA, AWG, human AWG accomplished is profound. And why don't you take it away?
Yes, absolutely. Thank you, Ryan. And I'm very excited to be giving this talk today, in which I'm going to be talking about a recent publication, Feeding the Cosmos. Next slide. Which, as Ryan mentioned, it has been a collaboration between, very fun collaboration, if I may add, between many exciting groups, such as BioAstra and different analysis working groups that Sometimes, otherwise, we may not collaborate together. And the first hypothesis that we have is that space environments change how plants take up and retain nutrients in crops grown in orbit. And these losses may directly and can affect astronauts' dietary requirements. So the question that we have is, how does spaceflight affect plant nutritional content and astronaut nutrient requirements? So our system for personalizing nutrition in space starts by looking at what nutrients are in food grown on a space station and on different space stations, and combining that with detailed health and genetic information from each astronaut from different missions. So first, different groups and missions measure the concentration of different vitamins and minerals that were grown in crops in space, in different space stations. And then we figure out which important nutrients might be missing for astronauts who eat only this food or crop grown in space. And this information is then matched with each of the astronauts' own body and genes to see who might be most at risk for different shortages and diseases. So again, why does this matter? If astronauts don't get the right nutrients, they can develop health problems, and we don't want to see that on long-term missions where we're trying to achieve huge feats for humanity. So as we're about to see, next slide. We analyzed lettuce grown on two different stations. So on one side, we have lettuce grown on Tiangong 2 Space Lab, so Latuca sativa, that was grown for 27 days and on a 15.5 hour photo period. And on the other side, that's where we have lettuce grown on the International Space Station on veggie hardware. We have growth on veggie 1A, 1B, and 3A for 33 days on a 16-hour photo period. Click. There you go. So analysis of all the nutrients combined led to both the ground control and the orbit having a similar concentration of minerals and vitamins. However, if we look a tiny bit closer, we can see that at the individual components, some changes. Specifically, we can see drops on calcium, magnesium, and iron. Next slide. So looking at the numbers, we can see the mineral profiling of lettuce from Tiangong 2 and the ISS that shows they both show a consistent drop in calcium, magnesium, and iron content. For example, the calcium level in Tiangong lettuce grown dropped from 928 milligrams per kilogram in controls to 642 milligrams per kilogram in space grown samples. And magnesium dropped from 365 to 274 milligrams per kilogram. So, and the ISS grown lettuce actually had a very similar trend. So what this is meaning is that diets relying on space grown food fall short in these minerals, raising the risk of disorders such as osteoporosis, hypocalcemia, anemia, and different immune weaknesses, among many others. So, and these are just the diseases that are associated with nutrient deficiency in humans. So now let's imagine that the compound effect of both nutrient deficiency combined with the astronaut molecular response in space, and things get trickier and they get worse. Next slide. So again, on top of certain crops that are grown on low Earth orbit with potential deficiencies, especially in certain key minerals, we have to fully understand the molecular mechanisms of different genes important for nutrition of different astronauts to have a holistic view of true dangers of eating certain crops long-term on grown on the stations. So for that, we have data from different missions. On one side, we have the famous twin study that was a duration of 340 days. So for that, we have samples from ground, pre-flight, in-flight, and post-flight. PBMC single cells, published in Science in 2019. As well, we have the Inspiration 4 samples, sample size of 4, duration of 3 days. So we have pre-flight post-flight samples, as well as PBMC single cells. You can find more on the OSD 574. JAXA cell-free epigenome, sample size of 6, duration of 120 days. So there is pre-flight, in-flight, post-flight measurements, plasma cell-free RNA for those samples, OSD 570. So now that we have a little, so I'm going to tell you a tiny bit more in depth about the pipeline that we followed. So the compound effect of this nutrient deficiency space grown crops and microgravity. First, we have to identify what are the nutrients and minerals of interest, especially based on the production of those nutrients from crops. And then we determine the associated genes to that mineral or vitamin. And for that, we use the IM database or KEGG. And after that is mined, we look for the astronaut data for expression of those genes using SOMA browsers, open repositories, etc. And the last step is to determine the associated diseases for the changes in expression of such genes and reduce availability of those minerals or vitamins, and ideally propose countermeasures where available. Next slide. Sorry. So a little more in depth about the pipeline. So again, what we're seeing especially here in the twin study database is that the calcium metabolism really takes a hit in space. So levels of urinary calcium, osteocalcin, and other bone turnover markers shoot up while astronauts are in flight. And then they only partially settle back down after the return, as seen on the twin study data that I see on the left over here. At the same time, many of the genes that help regulate calcium transport, signaling, and bone creation aren't really working as they should and have complete misregulation, as you can see on the graph here on the right. And this really highlights why it's so important to make sure that astronauts get different nutrients such as calcium in their diet, and then to explore the other molecular ways to protect their bones. So approaches like adding more vitamin D or flavonoids could really help boost things such as the WNT signaling pathway and shore up bone health, especially when paired with smart genetic and nutritional strategies. So again, then next click. Calcium deficiency in plants is really going to exacerbate this relation of calcium-related processes in astronauts during extended missions. So again, we are seeing a process in which plants that are grown have a lower levels of calcium. And again, on the other side, we have humans that have misregulation of these calcium-related genes. And these seem to be almost irrespective of cell type and irrespective of flight, as observed in the I-4, twin study, and JAXA missions. So for this, we look at 163 different calcium genes on different missions by RNA-seq, and there were only 8 shared among them. And we highlight ASPH, RYR3, CXCR4, MCU, ITPR2, PLGC2, PRKCB, and STIM2. And it seems like really misregulation of these genes could lead to different diseases such as hypocalcemia, osteoporosis, and potentially worsening the effects of already calcium deficiency in space-grown foods. And then on the right, we look at the top 10 calcium genes from each of these different studies and missions. So, for example, one of the examples is in the JAXA dataset. We observe significant downregulation of WNT5, which specifically contributes to osteoblast differentiation, which is key to building bone tissue. So, downregulation of WNT5 can disrupt these pathways, leading to weaker bone structure and a higher risk of fractures, something that we definitely don't want in our astronauts in space. Next slide. So we observe again that while next click if you don't mind. Thank you. that while many genes tied to calcium signaling are misregulated in space, the specific genes and how they behave can vary between astronaut cell types and as well as space mission. For example, certain immune cells such as CD4, CD8, and B cells and lymphoid cells, they show unique patterns in their calcium pathway responses. This means that spaceflight influences each part of the immune and bone system a little bit different, depending on the cell and even the mission type. But the big picture is still clear. No matter which genes or cell type are most affected, all missions point towards the same risk. We have weakening of the immune system and increased bone loss. And these problems get even worse if astronauts were to eat space crops full time that are already low in calcium. So while the details shift again from one mission to the other, the research challenge is making sure that astronauts get enough calcium and other nutrients to support their immune and skeletal health during long journeys. Next slide. And again, mitochondria, obviously, especially vulnerable in space, as highlighted by many studies from different members of NASA Genelab in great publications. And the mitochondrial damage is now hypothesized to be a central reason why astronauts experience so many health challenges during missions, including problems with their immune system, muscle, bones, and even vision. So one major hypothesis that we're proposing and we are observing is that calcium is absolutely critical for mitochondrial health. It helps regulate ATP synthesis and also controls the production of reactive oxygen species, which are byproducts that can really damage cells if not kept in check. So for example, the space flight, here you see on the right, disrupts the normal activity of genes that control how mitochondria function and manages stress. In particular, several calcium-related genes such as SMDT1 and PRDX5 are much less active in astronauts during missions. PRDX5 acts as a powerful antioxidant in the mitochondria. It scavenges harmful reactive oxygen species, helps keep the cell energy production running smoothly, and protects the genetic material inside mitochondria. So when the system is thrown off by spaceflight, it can lead to massive problems with energy generation and more oxidative damage, which put astronauts at higher risk of cell injury and diseases such as neurodegeneration. Next click. So again, calcium-related mitochondrial dysregulation may play a role in spaceflight-induced oxidative stress. Next slide. So again, I have been talking for the last minutes or so about growing crops with the right minerals and vitamins is actually essential for astronaut health during space missions. But just producing these nutrient-rich foods, it really isn't enough. You also need to take into account how well these vitamins and minerals are actually absorbed in the space environment. It makes a big difference. So recent analysis of just a cell-free, next click, RNA data has revealed important details about how microgravity can affect the gut's ability to absorb nutrients. So spaceflight, as highlighted by these beautiful publications of our great colleagues Kromer and Akinsuji, spaceflight can disrupt gut health, making the intestine more leaky. So a condition which is called the leaky gut syndrome, which lets unwanted substances such as different viruses and bad bacteria pass more easily into the body and can lead to wide range health problems. Next slide. So as seen by many of our colleagues, such as Walter, space travel can make the gut leaky, meaning the intestines becomes less effective at keeping harmful substances out of the system. We can see that in this graph, where there is a downregulation of pretty much almost all or most of the intestinal related genes important for mineral and vitamin absorption. So again, maintaining next click. Yeah, maintaining a healthy gut barrier is important in space. and it can be influenced by vitamins such as A, B, and D, and different amino acids, and beneficial bacteria. So counteracting this leaky gut in astros may require growing and eating foods rich in these nutrients, plus supporting the good bacteria in the gut to help astronauts healthy on long-term missions. So what can we do about this? So future forward missions and nutritional strategies for long duration attempts and missions should really focus on robust countermeasures against these deficiencies that have been highlighted in different missions. So a key emphasis should be the development of resilient nutrition systems that combine different solutions. So one of them could be crop selection. So choose crops that are naturally rich in vital nutrients. Should be further chosen to preempt deficiency, especially for minerals most affected by spaceflight, such as calcium and magnesium. So we can see some of those crops, such as soybeans and dill. Thank you, Ryan. That should take a closer look for further missions. The next steps will be biofortification. So this involves the genetic and enhancing of different crops with high levels of essential minerals like calcium or vitamins. Next click. And the next one should be bioproduction, or as I like to call it, using plants as a factory, such as the new paper that has been released, production of parathyroid hormone in Fc fusion form to support bone health, which could be very important for long-term resupply from Earth when there's no possible. And the last one is supplementation, which especially it might not be viable for extended missions with little resupply. Next slide. So again, I just want to highlight all of the people that made this possible and very, very fun conversations. So this was a great effort between BioAstra again and different analysis working groups and all the authors that are pictured over here in this picture. We spent a great summer going through and piercing all of this data that we think ideally could set up a new pipeline and new pathways for different scientists and missions to explore for nutritional requirements on our next step forward in humanity. So thank you very much for listening, and I'll take any questions in the chat or wherever possible. Thank you, Ryan.
Thank you, Borja. That's amazing. So it was an honor to be involved in just even to touch all that stuff and help see it come to fruition. And I just remember when Walter came on board, it was such a logical step to have a gut biologist. So yeah, cheers and thank you.
It was very fun.
Yep. There we go. All right, is Brian? Oh, there is Brian. Let me highlight you, Brian. So now your mic should be live.
Tested. Can y'all hear me?
We hear you.
Excellent.
And now I'm about to
see if I can
share your deck.
Okay. Having some problems with the video.
All right.
Okay. So awesome. So we'll go ahead without video.
It's fine.
Excellent. So thank you, Ryan. I'll provide a quick high-level overview of our projects, and then we'll give some of our researchers a chance to discuss some recent updates. So next slide, please. So our project focuses on creating AI/ML models to characterize DNA damage in cells exposed to radiation. The main purpose is to understand radiation hazards in deep space, long-duration space travel. So out beyond the protection of Earth's magnetosphere, astronauts are exposed to galactic cosmic radiation. So designing algorithms and systems to assess cellular damage associated with exposure is critical to survival in space. So this work not only has relevance to space travel, but it's also beneficial to life on Earth by assessing, for example, occupational hazards faced by airline crews, X-ray techs, nuclear engineers, and medical imaging patients. So the dataset consists of about 77,000 images of soleus muscle tissue that was immunostained for 53BP1 protein and irradiated with either X-ray or iron particle radiation of varying doses. right? So the 53BP1 protein, it forms a foci around double-strand breaks that are a result of exposure to radiation, and it begins the DNA repair process. So these foci show up as bright dots in the images, and this data set is available on OSDR and the AWS data registry. So our team's first deliverable is a paper comparing various classifier architectures and their performance on the FE X-ray image classification task, and also a radiation dose prediction task. So this is a very rich data set, and we have several next steps kicking off, including analyzing the 9-image Z-stack. So the original images we're looking at are compressed images. It's all 9 compressed into a maximum intensity image. We're going to start looking at more of a 3D aspect here. We got some AI/ML model explainability efforts that are happening. And we also have some GWAS data that we haven't looked at yet, but we're hoping to be able to tie that to our DNA architectures and link gene variants to phenotypes of DNA damage response. So next slide, please. Excellent. So before I pass the mic, I wanted to show what some of these images look like. So ideal images of nuclei irradiated by iron particles look like the images here on the left side. You can see a clear line or tracks of foci where double-strand breaks occur. And on the left side of the image, we have X-ray. Sorry, I got that reversed. On the left side, we have iron, and on the right side, we have the X-ray, where it's more like a shotgun, like scattered random foci. So with that, this task is not as simple as classifying pictures of cats and dogs, and now Michael will discuss some of the challenges we encounter.
Michael, that's right. There's like, give me 1 second, Michael. Just gonna spotlight you. Go ahead, Michael.
There I am. Okay. Yes, thank you, Brian. So, yeah, before we start working with classifiers, I thought it would be a good idea to actually look at the actual data. And what you see here is different exposures with different, well, actually all 4-hour time period post-exposure. But the most important ones are the two in the top row, the first one and the second one, and the one in the bottom row on the left, because those are the ones that we carry on most often. And so what you see here is that on the y-axis I've plotted the proportion of cells, and on the x-axis I plotted the number of foci that were detected per nucleus, and that's after the judicious application of some background extraction and some other cutoff levels to properly detect them. And the upper right corner of each graph you see the number of images that were involved with it. And in the top left we see the sham irradiated mice. And this is only for C57 Black 6J male mice. There is basically the exact same pattern in female mice or the other mouse strains that were available. There are very, very slight differences that are not statistically significant. But you see here that even in sham irradiated mice, well, we have a lot of, 0.7 or 70% of the nuclei that we detect have no detectable foci. But about 10%, maybe 30% in total actually, have more than 0 foci. And that's going to be, a little problem. In the middle graph, top row, we see X-ray, 1 gray, 4-hour post-exposure time, and the number of not cells, nuclei that have no detectable foci is now 40%, significantly lower than the sham irradiated, what you would expect because X-rays will induce double-strand breaks that we detect over here. And the number of, or the proportion of nuclei with more, of 1 or more foci has, significantly increased. Now we go to the iron, the heavy particle, and as Brian already shown, shows a very distinct pattern of double-strand break induction. We see an even lower number of foci with no detectable foci, and even more with at least one of them. But note that there is a significant overlap between the semi-irradiated, the X-ray irradiated, and the ion particles exposed. So even though you can at random pull like 50 nuclei from all your images and get the correct classification, that's not going to be the ultimate goal. So, but somebody in the further on will tell you all about it, I suppose. If you can show me the next slide, Ryan. This is just an example of 25 at random nuclei for the two conditions over there. You see that in the semi-irradiated, maybe the bottom row, the middle one, might have a little, foci, but I'm not quite sure. Of course, I don't have the exact numbers here. But you can see in comparison with the X-ray irradiated, there is, definitely a lot of, if not most of them, have detectable foci in them. Although some of them don't, and that's what you expect. And next up is Shahab.
Go ahead, Shahab. You're unmuted.
Hello?
Yes, we hear you. Cheers, thank you.
Okay, that's great. Thank you all for having me. So hello everyone, thank you for having me. So my name is Shahab, and I'm a second year computer science student. And over the summer, I've been working under Dr. Frank's supervision as part of the Google DeepMind Ready Research Internship at the University of York. And just to start off with an introduction of what Dr. Frank has been looking at. So he first looked at this problem by testing a vision transformer base model on the BPS dataset. And the model has been uploaded onto Hugging Face, as you can see here. So that gave us like a strong starting point. And my part has been to carry that work forward, training newer transformer models and really focusing on interpretability and explainability. And the idea is not just to kind of get good results with the models, but to actually understand why the model is making its predictions. Next slide, please. Yeah, so the first thing I did was actually go back and train the vision transformer base model that Dr. Frank had started with. And I managed to push the accuracy up to 84%, which was already a nice step up from the 81% baseline showed earlier. After that, I trained some of the newer transformer models, like the date and SWIN models. And as you can see, they gave really solid results too. The date distilled had an 83% accuracy, while the SWIN performed the best with 86%. And this just shows us that there's real potential in exploring different transformers for this kind of data. Now, in terms of how I trained them, I just kept it like fairly accessible. I used Google Colab, and the Hugging Face trainer handled most of the training loop, and Torch Vision was mainly for the image prep. And so I wrapped all of this into kind of like a reproducible notebook pipeline. So you would load a data set, choose which model you want to train, and you would also get visualization metrics at the end. Next slide, please. Now, the second big piece of this work is explainability. So accuracy is important, but if the model says, this is an X-ray image without us knowing why, it's not very useful. So we implemented multiple explainability methods, such as attention heat maps from the transformers themselves, SHAP for pixel level attributions, and also GradCam. These would help us see which parts of the cell image the model focused on when making its prediction. The top image is an example of the SHAP maps, and the bottom one is just general heat maps, and I will touch more on them in the coming slides. So here are just some examples of the attention heat maps. On the left, you've got correctly predicted images, and on the right, you've got the incorrectly predicted images. So transformers sort of have an attention mechanism that tells us which part of the image they focus on most when making a decision. And by visualizing this, we get a heat map where bright areas show high attention. For example, the model concentrated, you can see in the top left image, where the radiation damage features are, which is pretty interesting to see. Like, the model is looking at these images where the radiation damage was actually around that image. And just a quick note, the zoomed section you see on the far right of each row is literally just the same image cropped in closer. It's not really showing like a deeper level of analysis, but there's definitely room to refine this further. Next slide, please. And similarly, here are some examples, but here we've got GradCam and SHAP. Starting with GradCam on the left, GradCam works by tracing gradients back through the model to highlight which areas were most important for the final class decision. So the red areas correspond to irradiated regions that the model relied on to predict the sample as either ion or X-ray. While on the right we've got SHAP, so it gives us kind of like a contribution score for each image region. Red would mean like a region pushed the model towards its prediction, while the blue pixels pushed the model against that prediction. Next slide, please. Yeah, so here I've got just a couple more examples of the SHAP maps, and just to give you a better feel for the variety of outputs. What's interesting with SHAP is that it doesn't just give you a heat map like attention, it actually breaks things down into contributions. So I'll just leave this up for a moment so you can see how the pattern changes between the ion and X-ray images. Sometimes it's quite clear, and other times it looks noisier, which is also telling us something about where the model might be struggling as well. Next slide, please. And also just a couple more GradCam images here as well. You can see that in some cases the highlighted regions line up quite well with where the radiation damage features are, which is really encouraging, but in other cases the highlights are like more scattered or less clear, and that tells us the model is struggling with certain examples. And one thing just to point out is that this isn't perfect yet. GradCam has given us a more detailed view than the raw attention maps, but it still has its limitations. Sometimes the overlays can look noisy or vague, so interpreting them isn't always straightforward. That's why we've been treating this as more of a starting point for exploring model interpretability rather than a final answer. So what's next from this? So this is still a work in progress. The first thing is just to finish up and to refine the pipelines. We also want to dig deeper into the transformer models themselves and specifically look at which attention layers are actually the most useful for this type of classification. Another direction is adding new state-of-the-art methods as they come out. And finally, something that is interesting is to look at uncertainty quantification. So instead of just saying this is X-ray or this is ion, the model would also tell us how confident it is in that decision. And yeah, so that's all from me. Thank you so much for having me and for listening. I'll hand it over now to Walt.
I'm afraid we're way over time. We still have, if we're going to even have a poster session, I know Brian, I was hoping all of your collaborators could speak, but maybe you could take this into like Q&A discussion, Walter. I'm sorry about that. Otherwise, because we still have 5 more speakers to get through in the next half hour. 5 or 6 speakers? Yeah, it's Kylie, Abdul, Elizabeth. We have 1, 2, 3, 4, 5. That's an hour. That would skip the poster sessions. Yeah, so let's. I'm super sorry, but we need to move on. I'm gonna have to ask the rest of the speakers, just in the interest of time, if we can actually try to cut your time down to more like 10 minutes. And so from here on out, I'm sorry about this, everyone. I think it's just, long day and all that. And my slides I no longer have access to. Kylie, I no longer have access to your slide deck that you uploaded. Huh. I still have Abdul Rauf's though. Something must have changed on the access. That's unfortunate. Huh. Well, Kylie, I'll work on that on the side, but I want to. Abdul Rauf, I believe I saw you here, right?
Yeah, I'm here.
Yeah.
If I can share my screen, that's okay.
I'd prefer to share the screen, unfortunately, but yeah. So if you and I'm also going to give you a 1 minute warning, up to 10 minutes. Sorry about that. Thank you so much. Go ahead.
Okay. Thank you, everyone. First of all, thank you to attending today. It's a great opportunity to meet all of you. My name is Abdurraouf, and today, on behalf of the AWG Exosome subgroup, I'm going to share with you the work that we're working on currently. I'll touch upon some of the results that we have, some of the future perspective as well. So the main focus of this work is to get kind of a biomarker that can reveal the stresses that spaceflight put in the human body. So as you know, the rate of the space flight increased dramatically in the last 2 decades or so, and these kind of flights really introduce a lot of stress to the human body, especially the radiation and the microgravity. The next slide, please. Yeah, and those kind of stresses really affecting every single organ in the body, as you already see in previous presentation, but more specific, it really affected the musculoskeletal decay and also affecting the immune system, the bioenergetic failures, and things related to the mitochondria. And if we want the missions to the space to be really successful, we need some kind of biomarker that enable us to take care of the health of the crew and the astronauts and so on, so that those missions get successful. The next one, please. So if we want kind of a biomarker, this biomarker need to have 3 main criteria. First of all, it need to be quick biomarker, it need to be non-invasive biomarker, and also it will be very good if this marker can give us kind of high resolution information. So instead of said this astronaut is healthy or unhealthy, it tell us at organ level. Let's say, for example, the heart is affected, but the kidney is okay, and so on. So we need some kind of these main criteria in our biomarker. And if we have such biomarker, that will give us kind of a real-time monitoring of the health of the crew, and also give us ability for a medical selection. So we can say, for example, this astronaut can go to this flight, and the other one have to go home and get some rest. So what is a candidate? The exosome is a good candidate. First of all, what is the exosome? Exosome is kind of a natural vessels that mediate intracellular communication between cells, and it's typically carrying DNA, RNA, sometimes signal molecule, and so on. But as I just mentioned, they fit all of the 3 criteria. First of all, they abundant in the circulation, and they are in the circulation, so we can use them as like non-invasive biomarker. And more specific, they containing information, specific information to its source cell. So it can potentially provide some specific information about certain organ that maybe have an issue because of the spaceflight stress. The next slide, please. So our main hypothesis here is that the spaceflight stressor really elicits significant changes in the circulating exosome, which serve as like sensitive biomarker for different physiological stresses, inter-organ communication, and also the tissue remodeling. This exosomal signature will inform the risk assessment and also will guide the development of effective countermeasures. Yeah, so here's our workflow. We'll start with different studies that have astronauts, and then those studies, they take the sample from the astronauts, they make kind of multi-omics analysis. We'll discuss in details what they did, and after that, the next slide. Yeah, we will make enriched analysis based on the data that we collected from those studies, and finally we'll make kind of enriched target organs. As I said, we just like focus high resolution to see which organ get affected with which pathways. So the next one. So we rely in Genelab. We have 3 studies: OCD 481, 483, and 484. The next slide. The first study starts with just 3 samples. It's the small one of them, and they have 2 data points: 10 days before the launch and 3 days after landing. They extracted the blood. From the blood, they extracted the plasma, and then from the plasma, they have the exosome. They have the cargo from the exosome, and they make RNA-seq. The next one. The next study is just exactly the same setup, but they have much bigger number, 42 astronauts. They have 10 days before the launch, 3 days after landing, and the last data point they have in the return day itself. The same setup, plasma, exosome from the plasma, they get the cargo and they make RNA sequencing. First of all, why we repeat their own analysis? They already make the analysis because they only focus in one certain type of the RNA. So the first study focused in long-randing RNA, and the second study focused in the microRNA, and also both of the study have very bad quality. So here the setup that we use I use kind of typical RNA-seq. I make like automated RNA-seq flow that take the sample and run all of that very quickly in our HPC. So starting with downloading the data, we have FastQ to check the quality. We have tons of trimming and tons of cut adapter to make sure the data have a high quality because the quality was very bad. Then we're using star for the alignment, and after that we're using a tool called feature count to count the different feature. And finally we're using a pipeline in R called EdgeR to have differentially expressed gene in those studies. And finally we make the enriched analysis. So the next one, yeah. Here are the results that we get from the first study. When we put the cutoff as a p-value of 0.01 and the fold change to be 2 or more, we identified like 287. And the same study, when we just relax the p-value a little bit to be 0.05, we get 737. The next slide. Yeah. In the next slide, we get much larger number, which makes sense because if you remember, these have much number of asteroids. We get 641 and p-value 0.01 and 1,600 with a p-value of 5. But remember, we need a biomarker, so we take the common between them. We need the common digs between the two studies because we wanted to focus on things that happen in every single flight, not something like certain flight specific or certain condition specific. So we will take the comment between them and then we use gProfiler. So all of the data after that will be from the gProfiler. First of all, when you look to the plot from the gProfiler, it takes from you list of genes and then search against all of the database that you determine. Things like gene ontology, like KEGG, the ectome database, etc. And as you see, there is a very high cutoff in the top, which is adjusted p-value very, very low, but we still have result at that very high p-value, more than 200 result, which means the data is really, really significantly strong. The next one, please. So I will show you very quickly, like different example of the data that we get. When we make the analysis against wiki pathways, which have a lot of pathways, you will find that a lot of pathways are enriched, but in the top of them things related, first of all, to the muscle, to the bone, and also to different immunological pathways. The next one, please. Yeah, we also look to human pathology database and we find like a lot of abnormalities, again, especially in the immune system, in the bone and in the muscle, and different other tissues get affected too. The next one. Here we look to the gene ontology. We wanted to see which regulation, because remember this exosome is actually working as communication between cells. So we want to know which processes those exosomes have information to control. And we found a lot of interesting one related to this communication between different tissue. The main limitation here, although they give you a lot of information, doesn't tell you which tissue it's specific to. And we'll discuss that in a minute. The next one, please. Then we look to the human protein atlas. This is the interesting one because it tells you which tissue the protein of this gene is actually enriched in. So we will go back to this one after we finalize all of our analysis to make kind of enriched organ specific. Yeah, the next one, please. Yeah, and then reactome database, again, you will see in the top of them, most of the pathways is related either to the bone, to the muscle, or immune cells abnormalities. And in the KEGG, it is the same. You will see in the top, we have rheumatoid arthritis, IL-17 signaling pathway, and several other immune-related things. Yeah. Now, what we are working on right now, the third study, it have the same setup, but it's interesting because it have something different. And instead of take the plasma directly and check their RNA, they have the plasma, they put it in cell line, they using cardiomyocyte cells, and then see the effect in the cardiomyocyte cell itself, so see the communication effect itself, and also they using microarray instead of RNA-seq. This is something we are working on currently. The next one. We're also making a comprehensive search now. We wanted to create a comprehensive data set containing 10 enriched pathways that we're interested in. So we will compare the result that we get it from the digs to this enriched database. And finally, we also try to, from this enriched pathway data set that we created, to make it organ specific so we can come back to it. And yeah, the next one. Finally, we're also aiming to have, instead of just RNA-seq, we have multi-omics-seq. We were searching about proteomics data, combine the result to have something more powerful than just RNA-based. Yeah, that's all. Thank you. I try to finish in time.
Outstanding. Outstanding.
Yes.
Thank you. So great to hear the update from your group.
Yeah, thank you. I appreciate it.
Cheers. Let me yeah, and hope you can have some chat with some of the people on more discussion about it. So thanks for, and I'm sorry everyone about having to sort of, I just want to make sure that all the talks get their proper due, you know what I mean? So sorry about the timing, but next up is Kylie. Welcome, Kylie. Hi.
Hello, thank you. Can everyone hear me?
I can hear you, so probably yes.
Okay, perfect. Well, all right, let's get started. Hello, everyone. My name is Kylie Brahma, and I'm really excited to share with you today a project I've gotten the opportunity to work on as part of my time at NASA Ames Research Center this summer under my mentor Jessica Lee and with my project partner Austin Wellness. Together, we worked on the Space, Physical, and Life Sciences Archive of Scientific Hardware, or SPLASH database. And today I'm going to be going over an overview of SPLASH, its goals, its features, its current efforts, as well as discuss my time working on the project throughout the summer, where we started, our methodology, and the results. So without further ado, let's get started. Alrighty, so for a general overview, SPLASH is a new database under the Open Science Data Repository, or OSDR. It aims to provide the public with a searchable database that describes hardware for spaceflight biology experiments, both past and current, through two main ways. First, the database provides a centralized location that users can reference to gain a better foundation or better knowledge of the kinds of hardware and experiments that are currently or already have been done. Secondly, by providing this information, SPLASH also helps facilitate the design of new experiments and hardware as it provides users, researchers, and developers with a comprehensive understanding of what capabilities already exist and to expand upon. The result is that SPLASH provides both metadata context for its users and also helps shape the future for biological and physical sciences spaceflight hardware. Next slide, please. That being said, you may be wondering, what exactly does SPLASH do? And what does it include? Well, SPLASH has specific criteria for its instruments. Most notably, SPLASH aims to include instruments of a TRL of 8 or 9, meaning that the instruments themselves have to already be flown or are ready to be flown. And they have to be relevant to the biological and physical sciences divisions. These instruments can be from any era, any provider, and on any platform, meaning that instruments from both legacy and current spaceflight missions, any provider like NASA, commercial, academic, or other spaceflight agencies, or on any platform, whether that be ISS, free flyer, lunar landers, or suborbital, do apply. So outside of this criteria, SPLASH entries will also include a variety of features, including data on instrument technical specifications, links to OSDR payload entries, and relevant NASA Space Life Sciences Library literature. And it will also include the ability to contact PSCs to learn more about various instruments outside of what's currently on the database. And we are projected to release SPLASH to the public in December of this year. Next slide, please. As we get closer to this release, I now want to take some time to talk about where SPLASH was and all the changes that it's gone through since the beginning of the summer. At the start of the summer, the old version of SPLASH was in a very different state than it is now. During this time, the old SPLASH database contained 60 instruments, which were about 6 years out of date and included specifications with varying measurement standards. This old version of SPLASH was also only accessible to Ames VPN users and was not even accessible across other NASA centers. Next slide. So when my project partner, Austin, and I inherited the database, there was many areas that we wanted to develop. Specifically, we wanted to update current instruments and add new instruments to the database to provide a more comprehensive view of the instruments currently on there, and we did this through many steps. First, we collected all instruments into a master Excel sheet, which you can kind of see here. Then we divided the work, updating and adding new instruments simultaneously by gathering information for each instrument by utilizing websites, organization contacts, and NASA SMEs to provide as much up-to-date and relevant information for each instrument as possible. Next slide, please. The result at the end of the summer was that these 63 entries that were already part of the old database were updated and 84 new entries were added. And the new SPLASH database in total included over 120 instruments, almost doubling from where it had been before. There was also a user guide and manual that was created for users pending the December release, and over 10 plus instruments were linked to OSDR. Furthermore, alongside these features we discussed earlier, we assembled a list of improvements that our software team is currently focused on implementing. These include, next slide please, reclassifying the research section of the instrument page, creating a field for relevant or similar instruments, adding adding and implementing that point of contact field that was mentioned previously, and also adding a user feedback section so then users and researchers could discuss and review instruments, provide context into possible successes, issues, or other areas of improvement they think that the instrument could need, all of which could give more context to users of the database who might be interested in using that instrument or learning more. Next slide, please. The result was a more robust database that included many key features that we feel will improve the experience and access for information for our users. And we actually did learn a lot along the way. We discovered early on that we wanted to optimize the database for various users, and part of that was including as much information as possible that could be relevant. During this time, we also learned the importance of time-independent fields, as time-dependent fields, for example, things like instrument age, would have to be updated yearly and put a high amount of workload on our software team and contributors. We had to figure out how to provide this information while also not causing the same work strain. We also learned about ontologies and how to categorize hardware to enable the related instrument field we just talked about. And it was through these discoveries that Splash was created. And once all of these, once the entire thing itself is released, we believe that its impact will be pretty significant, as it's one of the only tools that categorizes and centralizes space and life sciences and physical sciences hardware by providing information to its users, and that can really help reduce the amount of redundant development and also enable more advanced spaceflight initiatives in the future. Next slide, please. Finally, I wanted to take some time to give an example of both the old Splash webpage and provide context to the kind of information that Splash will provide. Splash will cosmetically look very similar to this, but it will have a little bit more information. In that, though, these 4 tabs that you kind of see circled are going to be there as well. There's 4 of them. It's General, Specifications, Research, and Logistics. General relays an overview of the instrument, including things like that age field that we just talked about. Specifications gets into the technical specs. Research discusses what areas of research the instrument is relevant to, and this used to be a yes or no question. And logistics discusses the amount or extent that human crew time is needed for instrument operation. Next slide. Splash will include all of these 4 tabs with many key updates, only which some are listed here. There's 61 fields for each instrument, so just for the sake of all of our eyes, I only included a few. The general field now has time-independent fields, with age being replaced with things like first year of launch and flight heritage. The specifications are now all in standard metric units. The research section has been reworked to indicate whether the instrument conducts research in the field as a primary function, labeled primary, secondarily performs the research in that field, which means that even though the instrument wasn't technically created for research in that field, it can be used for that, marked as secondary, or the research field is marked as none for having no relation to that research area. Finally, the logistics tab has also been updated to have a more standardized format. So, next slide. Now, I want to take the last few minutes or so to discuss some of the current efforts that Splash is working on. First, in regards to content, we are continually adding new instruments and data to the database. And we're populating those links in OSDR and SLSL, and populating links to the similar instruments and related fields. Second, on the software side, we're finalizing this database for public release, integrating into the OSDR website, and making improvements to the user interface. At AWG, we are establishing a community, improving and curating content, standardizing that data entry, and providing structure for downstream use. And finally, we are planning on releasing Splash to the public at ASGSR, which is a conference coming up in early December. And we're releasing a request for information, or an RFI, to the community to get more input. Yeah. Finally, I'm trying to wrap this up as quickly as possible, but this concludes my presentation on Splash today. Thank you so much for listening and learning more. I really loved being part of this project, and I can't wait to see where it goes from here. Now, I just want to take some time really quick to acknowledge my project partner, Austin Wellness, as though even though he couldn't be here today, his work and contributions were integral to getting Splash to where it is now, and it really couldn't have been possible to do this without him. And finally, I want to thank you so much to our project mentor, Jessica Lee, who helped guide and instruct us through our time working on Splash and all of our crazy questions that we had to navigate her through. Thank you so much. And also thank you so much to the additional contributors who have aided in getting Splash to where it is and where it's going to be, especially those of the AWG hardware community. So thank you guys so much.
Outstanding. What a great contribution. Thank you, Kylie. I'm thrilled about
Yeah, thank you so much.
Yeah, thrilled about the hardware AWG and the Splash database. That's amazing. So
Yeah, thank you guys so much.
Yeah, make sure if anyone's interested to maybe chat with her on the side and
Yes.
yeah, continue the discussion. So next up is Elizabeth.
Yep, can you hear me all right?
Perfect. Oh, I didn't even have to highlight you or nothing. Yeah, 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 let me share full screen. Fantastic. All right. Go ahead.
Awesome. Okay, hi everyone. I'm Elizabeth Aislinger, and I'm super excited to talk today about spaceflight-related cellular senescence in the brain. So what are senescent cells, or SNCs? A lot of us refer to them as zombie cells. So their basic story is that they don't reproduce or function normally, but they refuse to die and wreak a lot of havoc until they're cleared. They also spit off nasty chemicals that can turn other cells around them into zombies too. So for a more formal definition, senescence is a state of permanent cell cycle arrest. They accumulate as we age and in the context of illness and environmental stressors, and they can also contribute to disease. They're associated with cell and organelle morphological alterations, apoptosis resistance, and importantly, the senescence-associated secretory phenotypes, or SASP. SASP is when senescent cells secrete inflammatory signaling molecules, which can help recruit immune cells to clear them, but it can also cause severe issues. They're highly heterogeneous, which makes their identification super challenging. Their gene expression, their SAS, their other traits vary based off the cell type, the tissue location, organ, disease context, and a bunch of other factors. Next slide. Senescence can have good and bad consequences. They are to some extent a natural phenomenon. We don't want cells that have severe damage to their DNA to continue replicating, and in that way they can work as tumor suppressors. At the same time, accumulation of these cells, especially ones with a more harmful secretory phenotype, can cause problems, including cancer. SNCs can also induce senescence in a paracrine or endocrine manner, often via SASP, and cause problems like protein misfolding, fibrosis, and inflammation. Drugs to clear senescent cells or modulate their SASP have shown promise in treating disease and extending lifespan, but to develop those treatments, we first have to characterize what senescence looks like so that we can know what cells to target in different contexts. Next slide. We know that spaceflight includes hazards that are known senescence inducers like radiation, and spaceflight even serves as an accelerated model of aging, so we can expect senescence to occur. And because of the unique environment in space, what senescence looks like and how it needs to be treated may differ from Earth. Next slide. For this study, we used a mouse brain single nucleus RNA sequencing data set, including younger and older spaceflight and ground control mice. We calculated cell level senescence scores using expression of canonical senescence genes. And since we often see about a 1% senescence abundance in healthy tissue, we took the top 1% of scores in the young ground control mice, and that defined the threshold for senescent cell classification in the other groups. We performed, next slide. We performed a conventional pre-processing pipeline and clustering using the overlap between cell type markers and canonical cell type markers in order to automatically label cell types, and then use ScanD for sample integration and label transfer. And then we cross-referenced automated annotation methods such as cell typist. And as you can kind of roughly see in the color patterns in the UMaps below, no need to squint to see plot labels for any plots. I'll cover any important information aloud. We achieved good consensus. Next slide. I, of course, also manually looked at the marker validation expression to validate. I know everyone here comes from different backgrounds, so not everyone will be familiar with all the cell types I talk about. But the key point to remember is that probing cell type differences helps us understand what consequences might be attributable to senescence, why they happen, and how to intervene. Next slide. For downstream analyses, we tested group differences in senescence scores and classification probabilities and whether that effect varied across cell types. We then performed composition analyses to detect group differences in senescent cell proportions among each cell type, and this is a different way of looking at abundance that uses Bayesian methods to take into account the compositional nature of the data. Lastly, we looked to see if certain canonical pathways were enriched in spaceflight. Next slide. As expected, we found spaceflight predicted higher scores and classifications, and it had a greater effect on oligodendrocyte precursor cell senescence scores. Oligodendrocytes are a type of glial cell that provides immune and metabolic support along with myelination, or in other words, the insulation that helps transmit neuroelectrical signals. And they're known to be super important in neurodegenerative diseases. Next slide. In aged spaceflight mice, we found increased senescence burden measured by the composition analyses in those OPCs, the oligodendrocyte precursors, as well as in astrocytes. And in spaceflight versus ground control, saw increases in oligodendrocytes and their precursors. In aged mice in both conditions, we saw increased microglial senescence. Next slide. Okay, moving on to gene expression. Here's, actually we can skip this slide. Next slide. More formally, I ran decoupler ULM on the data subsetted to just the senescent cells to find pathways enriched in spaceflight. In senescent neurons, those pathways included oxidative phosphorylation, reactive oxygen species, hypoxia, unfolded protein response pathways, and a bunch of others I don't have time to go over. When we further broke this down by condition and age, we saw certain signaling pathways that differed across age groups, which could foreshadow age-dependent outcomes and effectiveness of interventions. In oligodendrocyte senescent cells, we likewise saw enrichment of OXPHOS and ROS, and also xenobiotic and fatty acid metabolism and cholesterol homeostasis. In senescent spaceflight OPCs, microglia and astrocytes, we again saw OXPHOS enrichment, and in OPC specifically, inflammatory signaling pathways. Next slide. So to wrap up, we saw evidence of spaceflight conferred oligodendrocyte senescence vulnerability. We also saw the importance of oxidative phosphorylation and stress, which Drs. Fernandez and Pavlitish talked about in their excellent viral reactivation presentation, along with other pathways that might help us link spaceflight exposures to senescence mechanisms and their consequences, which could inform countermeasure development and even translation to Earth medicine. Next slide. So for future directions, like I mentioned earlier, identifying senescent cells is an enormous challenge. So it would be ideal to incorporate immunofluorescence or enzymatic assays to achieve some sort of consensus classification. Perturbation experiments could be helpful for intervention development, such as gene or immune therapies, or other treatments to reduce senescent cell accumulation or modulate their phenotypes to be less harmful. For example, Dr. Barbaro discussed the importance of nutrition and ROS regulation earlier. The gut-brain connection is another area I hope to probe further. I also have a gastroenterology background, as well as take inspiration from the wonderful work of Brian Everts and others on the DNA damage subgroup. We also hope to perform additional replications and extensions. So far, I did take a radiation data set and another OSD data set and found some promising convergence. And for acknowledgements, I want to thank NASA Genelab and OSDR, and especially our fearless leader, Ryan. None of this work would be possible without you guys. You're amazing. And of course, the Brain AWG members and my co-authors, Wendy McNerney, David Pekarsky, Anisha Pandey, and Nilofar Ali. Feel free to come find me later or reach out using the contact information to the right, and thank you for listening.
Whoa, another outstanding talk. Elizabeth, I don't think I've been following oligodendrocyte research for some time in space, but that's just some interesting discoveries that I don't believe were characterized before. So I need to marinate a little bit more and go back and check out the research. There was an oligodendrocyte ISS study several years ago. I'd have to go back and double check it, but a mental note to come back to that, but we do need to move on. So yes, everyone, please take advantage of everyone here to talk to one another. Yeah, that was just so good. Yeah, everyone, this series of talks are just outstanding. Julia, welcome. Hi, Julia. Okay, I'm got your deck. And yeah, thank you, Elizabeth, for helping us catch up on time as well. I just want to make sure everyone, all the speakers, feel equally, time allotted. So please, Julia, and of course Amanda too, but very, very happy that you were able to present today on the NASA Edge Bioinformatics platform. Go ahead. Oh, I don't think you're highlighted. I need to do that. Whoops. And spotlight. Now you're good.
Can you hear me?
Yes.
Okay, awesome. I can definitely speed talk to keep us on time.
No worries.
So I think I'll just acknowledge Amanda again. None of this would be possible without her. Just a dream collaborator in every way. So that's it for the first one. Next slide. Okay, so I have been a part of the microbes and the multi-omics AWGs for several years now, and then through these AWGs we were able to connect the Los Alamos National Lab, where I work, and the National Microbiome Data Collaborative, where I'm also the community engagement team lead with NASA Genelab. So from these meetings, in my capacity as a Los Alamos scientist, was able to connect with Amanda and some other wonderful folks at NASA Genelab, and with Ryan, and then also with many AWG members. And so then since then I've met and worked with many OSDR users, and a lot of what both Amanda and I were hearing is that there's an ongoing need across multiple AWGs for accessible options for running standardized bioinformatics workflows for multiple data types, and that this is really necessary for this whole moon and Mars, everything that we want to do, and anything biological. Yeah, go ahead, Ryan. So at Los Alamos, my PI that I've worked with for 8 years now at Los Alamos is Patrick Chain. So his group developed the Edge Bioinformatics platform. So this is an open source, user-friendly, free, accessible GUI for running bioinformatics workflows on sequencing data. It uses our sources, so it's really point and click, and the goal is to make it accessible and make bioinformatics easy for everyone. And we've hosted global workshops for this platform, so different groups across the world and in the United States to launch local edge informatics instances, as well as running on this to help global missions. And then we've had several spin-offs. So NMDC Edge runs the NMDC standardized bioinformatics workflows, and then we have Edge for COVID-19. We have one with the CDC. We've had different iterations. If you go to the next slide. So Amanda also came to these chats that we started having and was talking about these incredible Genelab standard pipelines and the workflows that NASA Genelab, the team, has been developing. So Genelab, if you aren't aware, works with AWG members to develop community-driven standardized pipelines. So the data processing team then wraps those pipelines into full workflows, uses them to reprocess omics data hosted on OS. So then processed data products are made publicly available on OSDR, which a lot of people have talked about today. So although all these Genelab pipelines and workflows are publicly available on GitHub, using them does require access to computational resources and some knowledge of how to run workflow and how to install and run these tools. So we found that this was kind of a perfect partnership where we had the infrastructure for the GUI side, they had these amazing workflows. So the Edge platform is that way to kind of remove those barriers and maximizing access to these amazing standardized workflow. Next slide. So the thing that we've been talking about is called NASA Edge. Very creative with our naming, I know. We just keep naming everything something Edge. But so Amanda and I worked really hard to present a proof of concept, and Ryan was there as well, of this preliminary version of NASA Edge at an AGU a couple years ago. And we used this to really gauge interest with the community as well. And from there, also talking to other AWG members, they were saying, yes, I would absolutely use this to help with the work that we're doing AWGs to process this data, work with these data sets, and turn more of this reused data into more publications. So we actually got a proposal funded from NASA in 2024 to develop this NASA Edge platform, and we got it to the proposal to add multiple workflows, develop some training materials, and all of this is in this Nextflow workflow implementation, which worked best for both of our teams because that's something that we wanted to pivot to in our Edge Bioinformatics platform anyway. So we do have some links here that anyone can access, but I'll talk a little bit about that in a minute too. So next slide. So the first workflow that we've been working on implementing is the Amplicon workflow. So again, Amanda has really led the charge on all of this, and then Alexis Torres on the NASA side as well has been told to this development. So we're starting with this Amplicon workflow for 16S, 18S, or ITS, which is wonderful for me also as a mycologist. I love to see the representation. So it's really a point and click workflow. So you can change some parameters as you can see in there, but it's really easy to understand. Everything is in there for you to run it, and you put in your data, you point, click, hit submit, and it's very easy to understand, hopefully, and easy to run. And one feature that we are about is you can actually directly grab OSDR data just with an accession number, so you can directly pull that over, and you can also upload your own files. So if you have your own files, your own data that you're looking to maybe compare against some OSDR data sets, or you're just looking to run your own Amplicon data, you can do either one of those. Just the uploading your own files does require a run sheet, as you'll see in the platform. Next slide. So Amanda put these nice visualizations in as well. So we have some amazing outputs, which is we've seen from other Edge platforms, that we always get the request for more visualizations, publication-ready figures that you can copy and paste onto a poster easily. So we're really excited to have these. We have visualizations for alpha diversity, beta diversity, taxonomy plots, rarefaction curves, dendrograms, and kind of things that we've heard from the users over the years that would be most useful to them. And all of these are automatically generated when you run the workflow. So we give you those raw downloadable outputs, but we also give you these interactive visualizations. This is just a continuation, so we also can do differential abundance analyses, and I encourage all the visualizations that we have available on the NASA Edge platform, because again, this is something that we're really excited about. Next slide. And Amanda has put in a ton of work to make user guides for NASA Edge and the Amplicon workflow thus far. So there's this wonderful documentation site and these tutorials. So there's some user guides, and video tutorials will be made available to all users, along with those static how-to guides to users through how to process both those OSDR data sets and the non-OSD data sets, or your own data. So if you want to use those workflows in this platform, I would definitely recommend checking out the documentation in these user guides. And yes, perfect. These are the user guides. So here's a little sneak preview. Next slide. Future plans. So we intend to be extremely responsive to user feedback in our user research rounds. We will also develop those additional training materials. We're hoping to run some workshops and hopefully expand the use from the AWG. So we are here also presenting this in its beta version to all of you as potential users. So we'd love to see you use this platform to process and work with the data for your AWG work. And then we did write in, as we said, all of these additional workflows that will be implemented into NASA Edge in the upcoming couple of years. And again, maybe thinking about what else would you like to see. We have been notorious for listening to user feedback for our other Edge applications, so we really want to know what you want to see in this platform. Next slide, please. Okay, and then finally, we are recruiting beta testers. So I know it's late in the day, but if you want to take out your phone and get involved here, we would love to have to beta test. So we'd love to have as many AWG members across these different groups sign up to be beta testers. You can have any level of bioinformatics experience. We're especially interested in getting novice bioinformaticians to see how accessible it really is. Your feedback will be anonymous, so we're collecting in this sign-up form. We are collecting some of your info, just like your email, so that we can email you when we open up the round, but your feedback will be anonymous. The Amplicon workflow will be available for beta testing probably around October 6th, be first, but we'll send out a full official email with the instructions in that timeline. So please sign up using this interest form. You can copy over this little link or scan the QR code. Yeah, and with that, we're just very excited to this is the first time that we're really presenting all of these new updates. So we're very excited, and I just want to acknowledge that this funding comes from NASA, from this ROSES grant, and then also the Los Alamos National Lab, Gene Lab, and then obviously Amanda, and then Alexis, and then Yan, Mark Flynn, Andre Watson, Patrick Chain, Chin-Chi Lo, and Bin Hu have been really instrumental. And these are just links to the Edge platform. The new updates that we showed you are in the development environment, which is where you would be beta testing, but then we do have this other live version that you might have seen before. But again, we're really excited to work with all the AWGs, and I'm excited to have this in the microbe and multi-omics AWGs that I've been a part of. And that's it. I think I kept to 10 minutes.
You rocked it. Thank you so much. I'm just so excited to see everything that's coming together on the Edge Bioinformatics platform, Julia, and Amanda, of course, and everything. So kudos to NASA and the funding and, NANL and everything. So cheers. And hopefully some of the microbes, AWG people especially, can come chat with you real quick and talk about getting even some more insider info and ideas on, maybe you're a grad student looking for like a microbiome related bioinformatic project. It's perfect, something to use for. So our last speaker is going to be Rachel. So, Rachel's already highlighted.
Can you hear me?
I can.
I'll try to be fast too.
I understand. We're just, and it just is. I mean, what an amazing day. Maybe we should have done. I don't know if we could have done a longer day. That's a lot of time. But my gosh, these are good presentations, so it's hard. Yeah, definitely. Last but not least, amongst our speakers. Rachel, from mostly out of the plant AWG. Why don't you take it away?
Thank you so much. Yeah, so I'm Rachel Rivero from the plant AWG, like Ryan said. I'm going to be giving a brief overview of one of our subgroups, as well as a related collaborative project with the plant AWG, and these are focused on understanding the challenges of regolith-grown plants and their context in human health. Next slide, please. I think we all here want to know how we will survive and explore beyond Leo, and of course, as the plant AWG, we want to know specifically how will we grow plants and therefore use them to help us accomplish life beyond Earth. It has been long proposed to understand if it's possible to grow plants in regolith. It's obviously an abundant resource, whether we're on the moon or Mars. It could reduce what we need to bring from Earth. It does have some nutrients, and it provides structural support for plants just like terrestrial substrates. And then there's the famous work showing that we can, in fact, grow plants in lunar regolith, but it isn't without very significant stress and physiological and growth costs. Next slide, please. So 2 points we want to look at, and as a follow-up also to Borja's presentation earlier at the start of this session, is how do we grow food that is nutritious while limiting what we need to bring with us, and how does spaceflight affect both human nutrition needs and plant growth? So looking at regolith studies is just one component of this, but the plant AWG has formed a regolith research subgroup led by Dr. Borja Barbera Barcinia, where we are working on a book of all plant growth experiments and regolith stimulants, as well as things like microbial associations, soil health, even looking at policy, and just the huge challenges of using regolith. In addition, the Moon Village Association's Lunar Agriculture and Sustainable Technologies, or LASS working group, is exploring plant elemental uptake and trying to connect it with human health challenges too. So this is also led by Borja and co-led by Harrison Coker and myself. And next slide, please. So to start with, and I'm sorry, it looks like the formatting's a little messed up here. But to start out with the regolith subgroup, we have determined chapters for the book, and we are starting with a collection of white papers. And I believe what we plan is to put them out into the community to get feedback on and build on, and that will turn into a book. So this is covering quite a diverse range of topics. So it's been really fun working with lots of subject matter experts on all of these topics and putting ideas together. So I just wanted to highlight a few of these things. So if you'll go to the next slide, please. One of the things we're doing is, of course, giving an introduction to space agriculture and life support, where we are at now, what has been done, how does regolith fit into the bigger life support systems, and where do we need to close the loop. But most of all, how does regolith fit into all of this? So that's the first chapter to start it out. Next slide, please. One of the others is composition, but even more than just composition of regolith, what areas do we have samples from on the moon? What do we know about geology, the origins of the moon? Next slide, please. These are just kind of fast.
Oh, it's amazing.
Also,
Yeah, go ahead.
also plant biology, just what are the stresses and growth costs, which plants grow best, what amendments, adjustments, and things like pH, fertilizers, compost are necessary. Basically, where is the field at and what do we know? Next slide, please. Another chapter is looking at how can plant microbe interactions be used to support this. It's a huge field in itself, and when we also have to consider how microbes themselves react to spaceflight, and even more, how do they act specifically with plants or in the harsh environment of regolith. But how can we use them to benefit plant growth in regolith? Next slide, please. We look at extreme environments to provide insights for engineering approaches, which organisms can support plant growth, which are adapted to specific environments, things like clean rooms shown on the left, low nutrient environments, things like this. Next slide. And then what do we do now about detoxification of regolith? How feasible is it to actually make it usable? So looking at some of the literature on that. Next slide.
Sorry. Yep, there we go.
And lastly, how can we consider regolith in line with automated growth environments to reduce crew time or even use it to learn more about plant growth? So these are some cool images taken by Harmeet, who I think he at least was here, but I'm sure he'll be happy to answer questions about some of these fun images. Next slide. And then the last working group, in collaboration with many Plant AWG members, is looking specifically at the moon. Since it is in collaboration with the Moon Village Association, it is looking at the moon, but it's specifically looking at a toxic uptake of metals and other elements from lunar regolith by plants. So can we eat those plants? Are these risks causing more stress to humans, to the astronauts living in an already harsh environment? And then linking these things back to support life on Earth as well. Next slide. One part of this work is building a global database of plant elemental uptake. So this has never actually been done. And so the hope is for this to be a proxy for lunar surface minerals. It contains so far over 100 studies with over 5,500 data points. It has concentrations of macronutrients, micronutrients, rare earth elements, toxic metals, and it spans all over the world. Next slide. So this will inform us, hopefully, how lunar plants may interact with surface minerals across the moon. So we can use GIS mapping of the high-resolution data that we have on Earth, like shown here in these pictures, and then relate it back to how the plants we have in studies are relating to the concentrations of these elements in the soil. And we hope to project that onto the moon, as I'll show in our plan later. And this will give us an idea of which plant groups, sorry if you'll go back again. So hopefully this will give us an idea of which plant groups might be the most productive in certain soils, which can withstand metals, things like that, and to get an idea of what might be toxic or deficient. And then now to the next slide. Oh yeah, looks like some of these are missing again, sorry about that. Just a transfer issue maybe. But we've also been looking at regolith simulant studies, which Harrison, Borja, and then also Angus have been working on. And we do see some greatly increased concentrations compared to controls. The ones that you can't see here are looking at particularly of importance, aluminum and copper, which showed quite high concentrations compared to the potting soil and terrestrial soil controls. And we want to know how can this affect astronaut health, right? So similar to the pipeline that Borja showed. So starting from the middle here at the bottom, we grow plants, they uptake heavy metals, and then does this have a toxic effect? Are those effects connected with diseases? We want to answer those questions. We're connecting elements with known astronaut response from 3 spaceflight studies and hope that this information can inform countermeasures like plant bioengineering and regolith amendments. Next slide, please. This one's quick because Borja already explained these studies, but we followed a similar pipeline combining data from these 3 studies, NASA Twin, SpaceX I-4, and then the JAXA Cell-Free Epigenome Study. Missing one again. It's okay. We then looked at some different time points in flight and compared them all back to pre-flight since we had that shared between studies. So it's very important to note that these are 3 very, very different studies, but thanks to OSDR and the SOMA browser, it's the data we have access to and provides some exploratory points for us to look at. So sorry if you'll go back again. We took the DEGs from each of these And then we pull genes that links to an element using the comparative toxicogenomics database. So at the bottom that's missing is a long list of elements, and then the number of genes that we pulled for each of those. So we want to use these genes to understand if there are diseases linked to some of these things. And interestingly, we found a huge increase in neurological diseases. So this is something that we're actually currently exploring and was hinted at in some previous presentations, but more in relation to toxicity. So stay tuned for some, hopefully some upcoming collaborations here. And then, so yeah, this is kind of layered, but we hope to combine all of the information and map it onto the moon and build a model of that. So with enough data, we want to determine the optimum site to grow plants on the moon. So at the bottom there was a lunar map, and then add elemental abundances, combine our predicted plant uptake from Earth and simulant studies, and then understand which elements pose the most risk and maybe at toxic levels, and then the diseases that might be related. And then the last slide, second to last slide. So I want to acknowledge all of the members of the last working group I've had the pleasure of working with, and who have been meeting to come up with these great ideas, and we've been having a lot of fun. And then the last slide, a special acknowledgement to Borja for leading the regular subgroup, and to Dr. Richard Barker for leading the Plant AWG, and of course a massive thank you to Ryan. I think everyone agrees that he's really the driving force behind this, and everyone else on the NASA side who's made this happen and supports the work that we're able to do here. So thank you so much.
Well, the honor is all mine, Rachel. I'm humbled at, I didn't quite understand the scope of what, I now see why you guys are working on a book, because you're sort of writing 20 papers, and it makes sense to
Yes,
sort of
we
put
are.
them together. I didn't quite get I didn't understand the full scope. So yeah, thank you so much. And that's the end of our Science Talks 2. To learn more on any of those, please reach out to those AWG members. And now, actually, the follow-on is our poster session. So if you go to the poster session A or B, depending on topic, you can follow up and hear some of the presentations, the poster presentations. And when you exit this room, you can go left or right, depending on which poster presentations you'd like to go to. So I know it's on my list, and I know there's plant, AIML, multi-omic, animal. There's a number of different presentations. So thank you, everybody, and that's the end of the main auditorium talks for today. So thank you, everyone. Well done, all. Cheers.